together somehow. We're gonna get it together and flower. We're gonna get it together. I know we're gonna get it together somehow. We're gonna get it together and flower. Up and up and up.
It's bad bitch o'clock, yeah, it's thick 30. I've been through a lot, but I'm still flirty. Is everybody back up in the building? It's been a minute, tell me how you healing. Cause I'm about to get into my feelings. How you feeling? How you feel right now? I've been so down and under pressure. I'm way too fine to be the stress, yeah. I'm not the girl I was or used to be. But you might be better. Turn up the music, turn down the lights. I got a feeling I'm gonna be alright, okay. Alright, it's about damn time. Turn up the
Okay, thank you very much. Good morning, everybody. Welcome for this fourth day of our 32 World Buiatrics Congress. It's a pleasure to be here. I am Ulises Bautista. I will moderate this room today in the area of short communications. So to start, we have a very great conference. It's by Professor Ali Fouladi. Sorry, let me just a second. Professor Fouladi, trained as a veterinarian in Tehran University, he gained his master's science degree in 1995. And, sorry, I have a little problems here, and PhD in 1999 from the University of Edinburgh in Scotland. He undertook the PhD studies at the Roslin Institute, where he worked alongside Professor Kate Campbell, creator of Dolly the cloned sheep, from differentiated cells. Following this, he moved on to undertake postdoctoral work at the University of Manchester before joining the University of Nottingham University as a research fellow, working with Professor Bob Webb to investigate the effects of diet on oocyte and embryo quality in lactating dairy cows. Joined the Royal Veterinary College as a lecturer in an animal reproduction 2006. He continued research on the impact of nutrition factors on fertility of female. His research is based on oocyte biology and embryology of domesticated and farm animal species and uterine embryo interactions and mechanisms of embryo implantation. So the title of this conference is Dietary Improvement of Fertility in Postpartum Dairy Cows. Thank you very much, Professor Ali. Welcome.
Good morning, everybody. Thank you for the organizer for the invitation. So let's move on. Go back. So I'm from the Royal Vet College in London. I'm going to talk about the nutritional improvements of the fertility in dairy cows. Better? Yeah. Okay, thank you. Right. Fertility of the dairy cows, as many of you have been perhaps familiar with this such figure, where the fertility of the cows reduced as a pregnancy rate to first service. That was because of the selecting cows for higher milk production. That trend basically was quite worrying, and lots of research has been done to understand that, and we see now a slight reversal in that trend. Otherwise, could be very damaging. So what I'm going to do today to understand why, as a result of why this decline is happening, and how we can use the nutritional management or managing nutrition to improve fertility in dairy cows. So for a cow to be productive and have a good fertility, they need to have a balanced level of energy that they consume through the diet, and to compensate for the maintenance, for calving, for milk production, and also fertility. However, when the energy level is low, they have or lower energy that they can receive through the diet, and that's not sufficient for all of that, so they go at the expense of reducing fertility. So, and that's because of the major changes that happens during the early lactation in the postpartum dairy cows, especially cows with the high milk production. And, I'll go back, sorry. here you see in the cells of the liver. Liver is you know, it is the basically center of metabolism in the body. And the cows, when they go to early lactation, they have reduced the glucose supply. So, and this, when they have energy through the reduced glucose supply, they have to utilize another way to produce energy. So they use this enzyme, it's called pyruvate and dihydroxy kinase 4 is upregulated, and through the electron transfer, and utilizes that to produce non-esterified fatty acids. However, this level of the NIFA increases, and NIFA we know is a toxic effect and causes the oxidative stress, and level of reactive oxygen species in the cells and the liver increases, and that causes the damage to the mitochondria. We call it mitophagy. And during this period of time, also we have limited supply of antioxidant systems in the body, including vitamin E and beta carotenes. So as a result of this damage, the cells undergo through the process of apoptosis, and it causes damage to DNA and the protein. And as a consequence of this, we have basically negative impact on the insulin signaling, which is blocked. And so that results in the level of the growth hormone, and the IGF-1 signaling, IGF-1 level goes down. So this could be causing a major issue. So here shows that, so during the early lactation, so we have the energy balance goes down, and NIFA level goes up. We have higher oxidative stress. So this oxidative stress causes the metabolic stress that can impact the follicogenesis, health of and function of ovarian follicles, and affecting ovulation, oocyte quality, oocyte maturation, and fertilization, can have basically negative impact on oocyte quality and embryo quality, pregnancy rate, and reducing fertility. So this is quite damaging from the beginning when we are going to have the consequence of that later on. Here the studies of my former PhD student, Walid in Antwerp, has done some studies on the granulosa cells of the ovarian follicles, basically on day week 8, but compare that association with the beta-carotene measured in the blood samples on day 2, and there are a number of 64 differentially expressed genes assessed by the RNA sequencing, and this is a number of pathways related to cellular response to lipids and ketones, immune response, all of them have been affected. So how does these changes can affect reproduction? So we have the high milk production, which can cause a negative energy balance, and then we cause a metabolic stress. This metabolic stress affecting metabolic hormones. We're talking about here insulin, IGF, and growth hormones. And when these levels are down, then affects the central nervous system, hypothalamus, anterior pituitary, affecting GnRH, FSH, and LH release, effect on the ovaries, which follicular growth, cyclicity, estrus, ovulation, luteal function, all of them have a negative impact. Also on the oviduct and the uterus, so fertilization, embryo growth, uterine health, and pregnancy, all of them are affected by these changes. And so why they are important? Because, sorry, I pressed this wrong. So because we know that the IGF and insulin are important both in the follicles and the corpus luteum for the process of the functions or synergizing with FSH and LH in the ovarian follicle, or synergize with the LH for production of the progesterone. And these two, estradiol and progesterone hormone, is important for function of the uterus to support embryos and reducing embryo loss. So in order to challenge that or use that nutritional factors, first you can basically enhance follicogenesis and increase more ovulations, or you can also use the basic diets which can influence the oocyte and embryo quality. So you can challenge that in two ways, and both of whatever you do could have impact on the endocrine profile of the animals, and also affects the uterine receptivity and environment that embryo needs to grow and establish pregnancy. So so I talked about the insulin and IGF-1 system. So this includes the IGF-1 and 2 and these receptors, and also IGF binding proteins. At least 6 of them are known that are involved in this process. But what is important is that for IGF to be active and help with FSH and LH system that I mentioned earlier, they need to be uncoupled from the IGF binding proteins. So IGF binding proteins, basically when they bind to their ligand, IGF-1 or IGF-2, they can inhibit their function. So in order for to have the bioactive IGF-1 or 2, they need to be uncoupled. Then they can bind to their receptor and do the function. So this is quite important for follicogenesis because during the follicogenesis, as the ovarian follicles develop, they become basically recruited and then FSH dependent, and they then become later stage LH dependent. So the IGF system has an important role in these stages. So, and that's quite basically important for the process of follicle selection for the ovulatory follicles. As the ovarian follicle progresses to become a dominant follicle and become ovulatory, we have a reduction in the IGF binding protein 2 and 4 mRNA, and that allows that the more bioactive IGF to become available, and that helps to improve the follicogenesis. So we know we have used this knowledge that, and I'm going to present some of the data related to that. So here shows the IGF binding protein 2 in the ovarian follicle. As you see here, when we come to the larger follicles, when they become dominant periovulatory follicles, the level of IGF binding protein 2 goes down. And that's basically was one of the reasons that the selection of the dominant follicle has been basically identified as the mechanism for that. So in the early experiments that Bob Webb and Carlos Gutierrez, that is one of the organizers here as well, was doing that during his work in Rosalind Institute. And we found that when increasing the plane of nutrition, twice maintenance affects the follicogenesis. So if you increase the energy that the cows receive, then you can enhance the growth of small follicles and medium-sized follicles. And this is associated with the reduction in the IGF binding protein 2 and 4. Okay? So that's resulted in increased level of the insulin. So here, after the experiment has been done with the twice maintenance or the basic maintenance diet, you see the level of the insulin in the circulation has been measured. And important finding here is the higher level of insulin at the time of basically ovulation or growth of ovulatory follicles. And related to that, you can see that here, as the follicles they grow to become ovulated, we have a higher level of insulin in the circulation. The same was for the IGF-1 in these cows, so they have higher IGF-1 in the circulation. So, and again, is related to the ovulation time. So this shows that if we increase the dietary energy, you can change the metabolic hormone concentrations, like here we talk about IGF-1 and insulin, and decrease IGF binding protein 2. That results in we have the increased bioactivity of IGF and increased sensitivity to gonadotropin and enhanced follicular growth response in the ovarian follicles and more ovulation. So in terms of the uterus, also has been shown that in the pregnant cows, they have basically the level of IGF binding protein 2 and 3 level basically goes down. And this results in the IGF that comes through the circulation, goes to the uterine gland, and they are available in the histotrough in the uterus, can support embryos, because the embryos, they have receptor for IGF, IGF-1, and then helps the embryos to grow. And so this works by my colleague, Claire Wattes at the RVC, showed that the cows that they become pregnant to first estrus or pregnant to the later estrus, they have higher level of IGF-1 in the circulation compared to the cows that they never become pregnant. So we use this knowledge so we say that if we increase the diet energy and or increasing insulin level, we can increase the follicogenesis. But what we have found that if we increase high insulin in the concentration, we have an effect on the oocyte quality. And that is something that we didn't expect at that time. And we found that so if we have give them cow high insulin diet or glucogenic diet, we have the number of blastocysts produced from these cows, from the IVF, significantly is dropped. And that was basically related to the quality of the eggs that has been done in this experiment. And in order to prove that as well, we looked at, I did some staining called differential staining combined with tunnel. So this differential staining shows the number of cells in the inner cell mass, we can count it, and the number of cells in the trophoblast cells. These cells, the pink cells, are making the placenta. Inner cell mass cells make the whole embryo. And the dead cells or damaged cells, cells that they go apoptosis, the green one, shows if the ratio of the inner cell mass to trophoectoderm cells is high, is indicating good quality embryos. And also, if the number of apoptotic cells in the embryo is high, it shows also the quality of embryo could be affected. So what we did, basically looked at the cells within the embryo. So despite the high insulin diet having higher number of cells in the total cell and trophoectoderm cells, but the ratio of the inner cell mass to trophoectoderm was much lower in the embryos from the high insulin diet. So the quality of embryos could be affected by that, the diet that was produced to that. So, so to summarize what I have said so far, so we can have the diets that can increase the insulin concentration, but increasing or improving ovarian activity, and then basically ovarian dynamics increases. However, this can have not be optimal for eggs as well. So eggs quality could be affected by the very high level of insulin. So in fact, they become like a diabetic condition. So alternative methods of providing energy for the cow was the dietary fat sources. Here in this experiment, we did feed the cows with the two level of the megalac, calcium super fatty acids, and they were fed for 2 weeks, and then we did the ovum pickup through the ultrasound, and then a large number of basically follicles were punctured, and as a result of this works, we have shown that the impact on the follicle dynamics and the oocyte and embryos that I'm showing to you here. What is happening here is that when we give them a high-fat diet or a low-fat diet, there are the distribution or population of the follicles changes. So low-fat diet, which had in fact high insulin, they have higher number of small and medium-sized ovarian follicles as compared to high-fat diet. And when we look at the oocyte and embryo development, here we say the cleavage rate in the high-fat diet is slightly higher than the high insulin diet. What the important and more critical is the blastocysts produced from IVF was significantly higher when produced high-fat diet or lower insulin. So insulin level is important, but very high seems to be damaging the egg, but it's good for the ovarian follicle, but it is good for oocytes. So we have more better quality blastocysts from these animals. So dietary fatty acids can reduce follicle numbers, but improve oocyte quality and blastocyst development. That was our conclusion from that part of the work. So based on that, we designed another set of experiments, which we give them basically high insulin diet or high-fat diet, and we call them cycling diet or mating diet. So mating diets improves the egg quality. Cycling diets enhances the follicogenesis. Okay, so this was the cross experiments. So a group of animals received cycling diet, then changed to the mating diet during basically after ovulation, and then we had the group of animals received mating diet all the way through, or received the cycling diet, then become a mating diet, and so on. So what was here was showing that here. So animals that receive cycling diet to help them to enhance follicogenesis at the beginning, then they have been given the mating diet to support the egg quality that resulted in significantly better results, as you saw here. So the cycling diet, a mating diet combination, they result in the basically over nearly 70% of the pregnancy rate until 120 days after parturition. So simply by the manipulation of the dietary energy sources, we can enhance or influence the ovarian follicogenesis and also at the same time improve the egg quality. One of the, when we talk about fats, so there are different sources of fats are available, and here we talk about the polyunsaturated fatty acids, which they are on different shapes and forms, more famous ones, we are talking about omega-6 or omega-3 fatty acids that you all have heard perhaps a lot. And these are most basically important members of each family. For omega-6 is the linoleic acid, which is 18 to omega-6. So here we have the difference between this group and that is the number of double bonds and the presence of the carbon, which on the first double bond is there. So, but there are, despite very high similarity between these fatty acids, but the functions of them are quite different. So here, in this experiment we did with Isa Dirande, and here we fed the animals with the same strategy that I mentioned earlier, with the 2 periods of diet. So first, period 1, to improve the phylogenesis, period 2 to improve the egg quality, okay? And here, give them the soya or linseed. Soya is the source of the omega-6 fatty acid. Linseed is the main source of the omega-3 fatty acids, or control here that they receive the palm oil. And what has been interesting here, the cows that they received, basically, for the first 40 days, they were receiving these diets. After that, then they shifted to receive either the controlled diet or linseed diet. So similar to what we have been discussing before. And the animals that received this combination diet and received more omega-3 diets, they have resulted in 82% pregnancy per AI. So significantly increased basically pregnancy rate. The outcome was much better in this group. And we did further some experiment more recently in sheep model and gave them different level of omega-6 to omega-3 fatty acids. And what we find that the level of this can affect the pregnancy rate and lambing rate in this. Not only that, can affect even the sex ratio of the fetuses. With the higher doses, we have more male to female. And I'll get back to you about this with some other results about the sexing of the offspring about that. So the high level of linoleic acid or omega-6 seems to reduce the pregnancy and lambing rate, whereas the omega-3 is improving the outcome. Other work that related to this, and I included in my talk, is about the another condition that happens during negative energy balance, which is the insulin resistance. It is a mechanism that the body basically applies to reduce supply of the glucose to reproductive system, but divert it to the other vital organs. And so in the peripheral blood, we have a reduced level of the glucose. And this insulin resistance basically challenging this or correcting insulin resistance through supplementing diets with some chemicals also was another focus of our study. And that one of the factors that we looked at is the PPAR gamma that we have basically applied. And this, in fact, there are this like a most famous one, pioglitazone, which binds to its receptors, and this binding to its receptor regulates the genes related to glucose and lipid metabolism, and as a result, we have the increased sensitivity to insulin. and that's in the liver, in the muscle, and even fat tissue. So what we did, we supplied or designed the diets which added pioglitazone, 6 milligram per kilogram body weight, starting from the 14 days prepartum, and in some groups, or in the, continued until 3 weeks after postpartum period in the PGT positive, whereas PGT positive negative received only during prepartum period, or PGT negative positive didn't receive at the early time, but then only received during postpartum, and negative didn't receive anything. So what happened was that here, you see there are, as a consequence of the treatment with this PPAR, we have reduced the NIFA. So NIFA level in the PGT positive was significantly reduced. And the same was the changes in the beta-hydroxy butyric acid. And there are reduced triglyceride, but important thing, there was the increasing the glucose, okay? Increasing glucose and also increasing the IGF-1. So that could be another strategy that could be used for challenging the negative energy balance period. And the same positive effect was found in the follicogenesis, and the cows, higher proportion of cows ovulated by day 14 postpartum, and they had a shorter open day. And the pregnancy by 120 days was basically higher than when we didn't have the pioglitazone. So 11% versus the 53%. Right. So in the second part of my talk, I talk a little bit about the oocytes. Here you have the ovarian follicles where the egg is located, and whatever we have high NIFA or low glucose, they can also affect the oocyte. In order to study changes in the oocyte, we need to understand the oocyte maturation. So we talk about oocyte nuclear maturation, where the oocyte changes during the pre-ovulatory period, goes from the germinal vesicle to metaphase 2 stage, where they are arrested and are ready for fertilization. Along with this, there are changes in the cytoplasm of the oocyte, including the mRNAs and proteins and other factors can support the embryo for the oocyte for the process of fertilization. Here, just go with some cartoon shape, which slide that to help you to understand the meiosis. The germinal vesicle is immature oocyte. When they go to, within 6 hours, they go to GVBD, or break germinal vesicle, and then progress to metaphase 1, okay? Where then we go to anaphase 1, then they go to telophase, and they go to metaphase 2, exclude the polar body, okay? But these are coming with the major changes in the within the oocyte. I'm not going through that because of time, but what is important, there are, when LH binds into a receptor for leading to ovulation, there are changes in the cumulus cells surrounding the oocytes. These are changes in the cyclic AMP and the MAP kinases, which are regulating the factors that influences oocyte maturation. And here we have MPF, or maturation promoting factor components within the oocyte, which they are phosphorylated. They need to be dephosphorylated to become mature oocyte. And these are influenced by the MAP kinases and cyclic AMP level. So what we did is the experiment in vitro, collecting the oocytes from the abattoir and treat them with the omega-6 or omega-3 fatty acids, and then looking at the cumulus cell expansion, and then health of the oocyte and oocyte maturation. So what we have found that the oocyte treated with the omega-6, they have a dose-dependent impact, a negative impact on the expansion of the cumulus cells. The cell death was higher in the higher doses. Okay, so even 50 microgram of omega-6 resulted in some of the cells dying, as shown by this green staining with the tunnel labeling. And here, with the omega-3 was different. So we have shown, except with a very high dose, we see some effect of that. And as you see here, in the cumulus expansion, was only with 200 micromolar concentration we see this effect. And, but the effect on oocyte maturation is quite interesting to see that with the omega-6 or linoleic acid has a significantly reducing oocyte maturation as compared to control. Whereas the omega-3, we have enhanced oocyte maturation, even slightly higher than the control. Remember that in the cow, oocyte maturation or IVF works very well. So just to test that we have added this concentration, we see they have enhanced the maturation rate. And even with the higher doses, some of them started to form the two-polar body, meaning they are hypermature and basically started to develop further. Look at the machinery in the cytoplasm with the MAP kinases, in this through the Western blot, and also through the changes in the cyclic AMP within the oocyte. You see the LA reduced the cyclic AMP level in the oocytes, whereas the ALA or LNA here increased the cyclic AMP within the oocytes. So there are not only nuclear maturation, the regulatory factor within the oocyte also was affected. The same was the MAP kinases. The LA reduced MAP kinase, ERK1, ERK2. Here, whereas the ALA has increased the MAP kinase. We have looked at the prostaglandin production by these cells, and we find that very high level of omega-6 basically produces very high concentration of prostaglandin in the culture medium, and that could be damaging for that. Quickly going through the changes in the mitochondria within the oocyte, we see the mitochondria in the oocyte, it basically regulates oocyte maturation. This staining with the mitochondrial marker shows when you see this cluster of mitochondria forming around the nucleus, that they provide energy to this oocyte that's essential for oocyte maturation. So oocyte initially in the GVA stage, they have more peripheral distribution of mitochondria. During maturation, this become more even distribution of in the oocytes. And when we add the LA or ALA, we see the differences in the distribution of the mitochondria, which is LA resulted in reducing that, and they have more remained higher in the immature way in the peripheral, whereas with ALA, we can gain the even distribution even 1 hour after culture. So that summarizes the impact of these two fatty acids on oocyte maturation and the regulatory factor between the two. Okay, so the final slides about the impact on the embryo development. Again, we see the LA as compared to the control. We have the reduced cleavage, was not significant, but the blastocyst rate was significantly reduced. Whereas with the ALA or LNA, we have the increased level of the increase the embryo production as well as the embryo quality, as was shown by this staining, the number of cells in total cell and the inner cell mass, and the ratio also was affected. So I mentioned that the impact on the sex of the offspring. Here we say that when we added this omega-3, omega-6, and also CLA is a conjugated linoleic acid, look at the outcome male to female. This is control 44%. This is following IVF, in vitro maturation and fertilization. When we added LA, the level significantly increased. So we have shifted more towards male. Okay, and that also was in vivo experiment where the animal received such diets. Again, we have the more basically male percentage increased. So, and the level of energy, if you give 3% protected fat or 5% protected fat, affected the offspring. And this staining was by, the detection was with the PCR, with the, identifying the presence of SRY gene, showing that they are male embryos. So to summarize as the text, I think I don't read that. I just leave that for the questions. If you have to, I will be happy to answer the questions. Thank you. Go back.
Thank you very much, Dr.
I had one. Thank you. Sorry.
Thank you very much, Dr. Ali. I think there is time just for a couple questions. Do you have anyone? Please, if you can go to the microphone, please.
Hi, good morning. Paula Gonzalez from Berbac, Australia. Have you studied the effect of chromium on improving insulin sensitivity?
Chromium?
Yep.
No, sorry, we didn't do that.
All right. That's right.
Thank you very much. Is there another question, please, Doctor? I think the last one. Thank you very much.
4 or 5 minutes.
In terms of the experiment in increasing insulin sensitivity, given that insulin resistance is important in driving lactogenesis, did you see a reduction in milk yield while you saw an improvement in reproductive performance? Did that result in reduced?
Interestingly, in that experiment, the milk yield was not affected. So that was quite encouraging for us as well. Thank you. I can provide you the data as well. If you want.
Thank you very much again. Please, let's give a round of applause again. And we would like to give you a present. Thank you very much for your presentation.
Thank you so much. Thank you for your time.
Thank you very much. So we are on time. The next speaker is Esteban Gallegos. Is now he. The title of the conference is Initial Report, Osteochondrosis dissecans emerging as an important lesion in postmortem exams in Mexico. Good morning, doctor. Are you ready? Oh, déjeme, sí.
This one? See? Good morning, everyone.
Sí, lo agradeceremos. Sobre todo para presentar mensajes un poco más claros del asunto. Is no problem if my presentation is in Spanish for you? Okay, thank you. I waited a minute for many people is looking for the translation, ¿no? Ok, esta mañana voy a dar la presentación realmente a nombre del doctor Rafael Ramírez. El doctor Rafael es uno de los más reconocidos patólogos que interactúan en los corrales de engorda en México. Él es un señor muy curioso y recientemente haciendo las visitas a los diferentes corrales de engorda o algunos corrales de engorda en México, pues con él hacemos, por supuesto, la práctica de la realización de las necropsias. El objetivo, perdón, y me fui para adelante. El objetivo de la presentación, pues, es mostrarles algunas lesiones que nos encontramos, como dice el doctor, de manera incidental al estarse realizando las necropsias de manera rutinaria. Esto fue en algunos corrales de engorda que en México, principalmente en las zonas del centro y norte del país. Se llevan más o menos 2 años de revisión una vez que se encontraron estas primeras lesiones sugestivas a esta condición, y eso nos ha dado más oportunidad de revisar y de encontrar este proceso. Como les mencionaba, en los últimos 2 años empezamos a tener esa preocupación por la gente que se encarga de la salud dentro del corral de engorda, sobre todo por un incremento en la manifestación de problemas que en México le llamamos podales, problemas de patas, problemas en los pies del animal. Esta preocupación fue bastante manifestada por la gente que está en las zonas de salud. Entonces, tratando de encontrar la causa del incremento de estos problemas podales, se empezaron a hacer las necropsias un poco más a detalle, y fue que se empezaron a revisar las articulaciones de los animales. Realmente, lo primero que estábamos buscando eran lesiones sugestivas al síndrome poliartrítico causado por micoplasma, debido a que constantemente encontrábamos lesiones en las necropsias sugestivas a una bronconeumonía supurativa relacionada completamente con micoplasma. Entonces pensábamos que de alguna manera el síndrome poliartrítico era lo que estuviera causando esas lesiones en las articulaciones. Otro dato curioso fue encontrarnos, como este no es el mío, otro dato curioso fue que encontrarnos en estos periodos de engorda, a la mitad del periodo de engorda y sobre todo en la finalización, la mayor manifestación de este problema podal. ¿Por qué nos preocupaba? Porque los animales en esos momentos de finalización pues tenían más problemas para caminar dentro del corral, era más alta la manifestación del problema. Entonces estos animales tenían menos oportunidad para competir por alimento al entregarse el alimento en el comedero, y eso ocasionaba retrasos en su desarrollo, retrasos en su incremento de masa muscular, y además había pues un problema que los iba debilitando bastante. Eso fue otra de las condiciones que nos empezó a llamar la atención. Como les menciono, fue durante los últimos 2 años donde fue aumentándose esta preocupación por la gente dentro de los corrales de engorda de esta manifestación. Entonces se empezaron a hacer las revisiones, principalmente en las articulaciones radiocarpales y tibiotarsales, como les menciono, realmente en busca de lo que probablemente era una condición causada por algunas bronconeumonías. Y por eso el doctor menciona que esta fue un hallazgo incidental, porque buscando las lesiones sugestivas a micoplasma fue que se empezó a reconocer una condición diferente en esa articulación, y nos empezó a llamar la atención esta situación, y fue que el doctor estableció el diagnóstico como una osteochondrosis. Ojo, la osteochondrosis no es la causa de muerte, no nos preocupa la osteochondrosis como un causal de muerte. Lo que nos preocupa es la alta incidencia, casi un 78% en las necropsias que se han realizado, es un dato que maneja ya el doctor como algo habitual. Más o menos un 78% de estas lesiones se presentan en los animales cuando se están revisando sus lesiones macroscópicas durante el proceso de necropsia, y eso es lo que nos ha estado llamando mucho la atención. Las causas principales de muerte pues, son bronconeumonías, acidosis ruminales, pericarditis, incluso fallas cardíacas. Algunas de estas relacionadas justamente por la inconsistencia en los patrones de consumo de alimentos, sobre todo las acidosis, pericarditis, algunas fallas cardíacas como consecuencia de neumonías intersticiales, también como consecuencia de una probable acidosis ruminal. Eso es lo que de pronto nos ha estado preocupando, que la manifestación de esta osteocondrosis aumente las mortalidades causadas por otras condiciones. Entonces, a lo que le queremos poner más atención es seguir observando si es que nos encontramos más esta problemática, y recomendar o observar o evaluar lo que está pasando con los animales en los sistemas de pastoreo. La gran mayoría de los animales llegan a los corrales de engorda procedentes del sureste mexicano. Son zonas donde los animales se están preparando en pastoreos previos. Esta condición de osteocondrosis, el doctor Rafael está seguro que es una situación crónica de una disminución o de un consumo bajo de los minerales que pueden ayudar a los animales a reforzar su sistema óseo. Debido a que en los últimos 2 años en México y prácticamente que en todo el mundo hemos tenido una sequía muy severa, pues se piensa o se asume que la calidad de los pastos en las regiones donde los animales están preparándose para luego llegar al corral de engorda no es de la mejor calidad. Entonces, por eso de pronto hemos encontrado esta deficiencia crónica de algunos de los minerales. Se ve más impactado en el corral de engorda justamente cuando el animal se está acercando a la finalización, debido a que el peso del animal está aumentando, estamos aumentando sus masas musculares, y llega algún momento en que el peso del animal más su condición de probable dolor o de debilidad en las articulaciones, pues no le permite al animal moverse adecuadamente dentro del corral como lo mencioné antes, para la competencia de la búsqueda del alimento. Eso fue otro punto que nos llamó mucho la atención, que conforme íbamos avanzando en encontrar este problema, los encargados de la detección de los enfermos en esos puntos, era muy común que nos dijeran, es que los animales se han pasado postrados un tiempo prolongado dentro del corral. Los vemos que están echados, no caminan, no se mueven. Al momento de nosotros estimularlos al movimiento, batallan para levantarse, se quedan mucho tiempo tratando de moverse apoyados en sus articulaciones, y se les nota que tienen dolor al levantarse y pues más dolor al caminar. Entonces ya cuando intentaban llegar al búnker, al comedero, ya era insuficiente para ellos la cantidad de alimento que se encontraban por ahí. Entonces, si ya tenían lesiones previas por algún proceso neumónico, y luego le podemos sumar algún proceso de acidosis ruminal por esta pérdida de patrón de consumo de alimento, es lo que nos preocupa, que esta condición de osteocondrosis pueda agravar algunas situaciones que realmente deberían de ser controladas. Y de pronto podemos estar tomando malas decisiones en los ajustes de los procesos de cuidado de los animales, porque lo que vemos es una alta incidencia de mortalidad. Y en ocasiones podemos tomar alguna decisión equivocada en donde tenemos que hacer los ajustes para corregir estas mortalidades. Cuando lo que pensamos es que de pronto un manejo previo adecuadamente, sobre todo de la suplementación mineral, puede ser un condicionante para la manifestación más severa de esta presentación. Creo que me voy a regresar unas 2 diapositivas porque me estaba perdiendo un poco. Sobre todo, no es cierto, más adelante. Sobre todo por estos puntos, otras 2 situaciones que nos estuvieron llamando también la atención en lo que nos encontrábamos en las necropsias. también durante los últimos 2 años hemos encontrado en un porcentaje más alto, tampoco es muy alto, un 4 o 5% más o menos en las necropsias, esta condición que conocemos como hematuria enzótica. La hematuria enzótica es el reflejo de que los animales tienen mala condición o mala calidad de pasto en donde están siendo preparados para la engorda. Y debido a que hay un mal crecimiento o una mala calidad del material vegetativo con el que tienen que ser alimentados, buscan hierbas, buscan plantas, buscan algunas otras fuentes que ellos ven verde ahí en el rancho. La hematuria enzótica es una manifestación de consumo alto de una planta que conocemos en México como el hecho macho, Telidium naquilinum es su nombre científico. Cuando el consumo de esta planta es crónico, ocasiona la manifestación de pequeñas tumoraciones en la zona de la vejiga, lo que causa obstrucción vesical. Cuando se nos ha estado incrementando esta manifestación de hematuria enzótica, es cuando también nos hace sospechar que los animales no están siendo satisfechos o en sus requerimientos nutrimentales debido a la mala calidad o el mal crecimiento de pasto ocasionado por la severa sequía que tenemos en México en los últimos años. Entonces, si le vamos sumando estas situaciones que nos indican que tenemos que hacer algo con la mejor aporte de nutrientes en los pastoreos, eso es lo que va a poder ayudar o a minimizar el que nos encontremos esta situación dentro del corral. Les digo que el doctor es curioso y también ha hecho el doctor histopatologías de glándula parótida. Al hacer histopatología en glándula parótida para el doctor es una manifestación clara de que ciertas condiciones que él encuentra en su estudio histopatológico indica una deficiencia de vitamina A. Otro condicionante que también nos puede decir que el consumo de betacarotenos, que son los precursores de la vitamina A en el pasto, está siendo no adecuadamente suplementado o consumido por el animal. De alguna manera los referentes que tenemos de la sequía prolongada, tal vez un crecimiento inadecuado del pasto y estas situaciones que nos encontramos, como bien dice el doctor, de manera incidental en las necropsias, pues nos hacen pensar que realmente las situaciones tanto de osteochondrosis como hematuria, como deficiencias de vitamina A, son consecuencias de un consumo inadecuado crónico o no cumplir con el requerimiento nutrimental que necesitan los animales previo a que lleguen al corral de engorda. Esto es una invitación a hacer esa revisión en los sistemas de pastoreo para ver cómo se están realizando la suplementación de los minerales, de las vitaminas, haciendo adecuadamente análisis bromatológicos del pasto que está disponible, determinando adecuadamente la carga vegetativa que puede soportar un terreno para ver cuánto ganado puedo estar pastoreando, etcétera. Todo lo que se debe de hacer previo al corral de engorda. En el corral lo único que estamos observando ya es la manifestación de una situación crónica. Pensamos que si esto se resuelve adecuadamente en los sistemas de pastoreo, pues va a ser mejor resuelta esta situación en el corral de engorda, debido a que a pesar de que se hacen los esfuerzos ya cuando el animal llega al corral de engorda de dar una adecuada suplementación mineral y vitamínica, pues de pronto el tiempo de estancia o la oportunidad que tienen los animales de consumo puede ser no suficiente para corregir el problema crónico que ya tienen. Eso es lo que nos preocupa. En el corral de engorda se puede hacer este esfuerzo por tener una adecuada suplementación, pero el daño crónico no puede ser tiempo suficiente dentro del corral para que se corrija. Entonces, la invitación es seguir haciendo investigaciones, seguir haciendo evaluaciones, como dice el doctor. Estas son las primeras incidencias que nos hemos encontrado. Ha sido, de pronto él dice, una casualidad. El dato que que da que el doctor, hablar de que el 78% de los animales que mueren dentro del corral de engorda tienen manifestaciones de este proceso de osteochondrosis, pues definitivamente habla de que es un proceso nutrimental previo al corral de engorda que debemos de estar corrigiendo. Se van a seguir haciendo estas evaluaciones para ver cómo es el incremento, el mantenimiento o la corrección de este problema. Estas son algunas fotografías de pequeñas lesiones, pequeñas sugerencias que al doctor fue el que le empezaron a llamar la atención. Algo que de pronto, si no le ponemos tanto detalle en la revisión a la necropsia, se nos pasa, se nos va, no buscamos de pronto estos detalles en la necropsia. Digo, afortunadamente que el doctor es muy curioso y empezamos a ver esto. El origen, repito, de revisar las articulaciones realmente fue encontrar lesiones sugestivas a micoplasma, pensando que había un síndrome poliartrítico por micoplasma que de pronto lo teníamos por ahí no adecuadamente detectado. Pero estas pequeñas manifestaciones de desarrollo inadecuado de cartílago y del tejido óseo que está alrededor fue lo que empezó a llamar más la atención. El doctor ha hecho sus histopatologías y ha demostrado que la calidad de los cartílagos, la calidad del tejido óseo alrededor de la articulación definitivamente no es la adecuada. Y es lo que puede estar causando este dolor, esta inflamación, esta manifestación clínica del animal ya dentro del corral. Hay muchas fotografías, muchas incidencias que de pronto hemos estado revisando y evaluando más constantemente. Les puedo mostrar ahorita de mi teléfono celular. El doctor no pudo acompañarnos, él está ahorita dando un taller de necropsias justamente en una zona del centro del país, en Querétaro, y ayer mismo me acaba de mandar 3 fotografías de esos animales que ha estado necropsiando con una condición igual o más severa que las fotos que les estoy mostrando, y es ganado que está en el centro del país, en Querétaro. 2 de los animales de las fotografías que me mandó ayer ya tenían prácticamente 120 días en engorda. La causa de muerte que determinó el doctor, de acuerdo al hallazgo de las lesiones macroscópicas, fue acidosis ruminal. Pero probablemente esta acidosis ruminal no fue tanto por problemas de la dieta, sino problemas del animal para poder tener un patrón de consumo adecuado debido al dolor que estas situaciones pueden causarle al momento de que él va a busca y tiene que pelear por alimento. Esa es la conclusión que da el doctor otra vez. Esta condición de osteochondrosis no es la causa de la muerte, pero puede condicionar a otras situaciones que puedan aumentar la probabilidad de que estos animales mueran por otras condiciones o minimizar la manifestación de otras probables enfermedades, sobre todo acidosis ruminal y neumonías intersticiales como consecuencia de este proceso. Entonces hay bastante incidencia, somos poco curiosos, e incluso hay corrales de engorda que la práctica de realizar una necropsia no la tienen como una rutina. Es otra invitación también a los que estamos dentro del corral de engorda que la necropsia la hagan realmente como un proceso rutinario, porque nos permite observar, encontrar realmente las causas donde podemos hacer las correcciones para minimizar las mortalidades. Lesiones bastante severas. Digo que ya cuando les ponemos atención, este hueso está muy poroso, entonces no sostiene bien el peso ya del animal conforme va creciendo dentro del corral, causa mucho dolor, mucha incomodidad del animal al caminar, entonces se mueve menos dentro del corral. Es curioso, pero sí me ajusté a mis 20 minutos, normalmente no lo hago, me paso. ¿Alguna duda, alguna pregunta? Les digo, es una primera observación, es una primera vez que se encuentran estas lesiones. Vamos a seguir poniéndole hincapié a esta situación, y conforme vaya avanzando tendremos más información.
Okay. Thank you very much, Dr. Esteban. I think there is no more time for any question. We are just on time. Thank you, doctor. So our next speaker is Dr. Swinkels. He's from Netherlands, and the title of the conference is Randomized a non-inferiority trial evaluating the efficacy of a novel teat sealant in pasture grazed dairy cows. Thank you very much, doctor.
Hello, everyone. I'm here speaking on behalf of my New Zealand colleagues that did this trial. Unfortunately, they were not able to come to the Congress, and they asked me to present their study that was comparing two teat sealants in New Zealand under the pasture-grazed dairy cows under the New Zealand circumstances. In New Zealand in 2021, MSD launched a novel teat sealant, which was a generic of the teat sealant that was the market-leading teat sealant on the market in New Zealand. And a generic, in principle, is exactly the same as the original product. And if you register a generic, you don't need to do clinical trials because it is assumed that the clinical results will be the same as the original product. But customers in New Zealand were saying, yes, but this novel teat sealant, investigational product, we will call it later, is not the same as the original product because you have less air in the tube. The viscosity is a little bit different. And so vets and farmers were asking, is it really the same? And if we said yes, they would, it was clear they needed proof that it was indeed exactly the same. And that is why we did this trial. And we did a non-inferiority trial, which is obvious if you compare two products. It's an obvious statistical method to say that you are the same or not. We compared the products on three aspects that are obvious. It's clinical mastitis in the next lactation. It's somatic cell count in the first herd test, 30 to 60 days in the next lactation. That is because New Zealand does herd testing only 4 times per year. And number 3 is the presence of teat sealant after calving. So those 3 criteria were compared. The non-inferiority test, we used very low margins. So here, you see for the clinical mastitis, you see a 2% margin, which is very low, because other studies, they were using margins of 10% or 5%. So we used a very low margin. The second one is the somatic cell count. We used also a very low margin of 10,000 cells. So if the difference would not be bigger than 10,000 cells, we would say it's the same. But if the difference was bigger than 10,000 cells, we would say that they were different. And the third one was the teat seal presence at calving at the first milking. And that margin was 5%, which is a little bit wider, but still very low. The farms were selected based on that they needed to be large, more than 500 cows. They needed to be willing to participate. They need to have suitable facilities. And they need to do herd testing 4 times per year. That's what they do in New Zealand. And they need to have treatment records, and those were the criteria that we selected the farms upon. Dry period in New Zealand is usually at least 2 months, sometimes even 4 months. So it's between 2 and 4 months because it's seasonal farming. Yeah, they are all dried off in the same period in the year. They had to have at least 3 functional quarters, body condition score bigger than 3. They should not have been lame, and the somatic cell count is, this is typical for New Zealand, is you're only allowed to, is for selective dry-off, the threshold is 150,000, below which you can only use a teat sealant, and above which you can add the antibiotic. So that is, and 120,000 for heifers. So 150,000 for older cows and 120,000 for heifers. So no clinical mastitis in the current lactation, positive pregnancy, and all cows needed to have treatment records. So the calculation of the number of cows, which is very important in a non-inferiority trial, because if you have low numbers, you'll find no difference. You're sure you find no difference. So in a non-inferiority trial, it's very important that you calculate accurately the sample size. For the teat seal retention, we assumed 90% of quarter retention, so that means that at calving, 90% of quarters would have teat sealant in it. And you used 80% power and margin of 5%, as I said. Then clinical mastitis, we assumed the incidence was 5%. We used that margin of 2%, what we discussed. And we included variation between farms and clustering within cows. And then here we would need 223 cows, and here we would need 533 cows. For somatic cell count, we assumed the log somatic cell count would be 3.7, and the margin would be 0.2, which is equal to the 10,000 cells that I just mentioned. So, well, you have different numbers, so we chose the highest number to be sure we had enough cows, and said we have to have 533 cows in each group. So this is the team in New Zealand, the combination of technicians and veterinarians that were doing the job. Yeah, of course, if you do a teat sealant trial, it's very important that you apply the teat sealant in a very hygienic way, not to cause any problems after teat seal, and to make a fair comparison. So what they did is they wiped and treated one teat at a time. So often you disinfect 4 teats and then you treat the 4 teats. But here it was done 1 teat, disinfection, and then treat. Then the next teat, disinfection, treat. So that's the way they did it. They were using 70% methylated spirits, and either teat wipes with that or cotton wool ball. And then they would disinfect the teat end until the wipe came away spotless. So if you saw a little bit of manure or dirt on the cotton wool or the wipe, you would disinfect again. So often they were disinfected 2 or 3 times to make sure that nothing was visible on the teat. There was partial insertion, a maximum of 3 millimeters with both tubes. That is important because it can influence new infection rate. No massage upward, and all teats were disinfected with chlorhexidine after infusion. So they first, when they were on farm, they threw a coin, and when it was, that's how they started. They put, if it was tails, they used the control product, and if it was heads, they started with the investigational product. And then alternated in the rotary, because New Zealand cows are milked in rotaries. They have no barns. They're always on pasture, and they get milked in rotaries, and all interventions are usually done in rotaries. So also the dry-off is done in rotaries. The cows that were dried off with the investigational product got a blue leg band, and the control product a yellow leg band. Therefore, the ones that applied the product were not blinded because it's clearly visible. But the farm staff and the data analyzers were blinded because they didn't know what those colors meant. Sometimes a cow would drop off in the rotary, something went wrong, and then they would skip that cow, and then they would continue the alternation. So if you use the investigational product and the cow drops off, you would simply do the next cow with the investigational product and then continue. So that's how they did it. If you dry off 1,000 cows in thousands of cows in a few days, that's of course, this is then the harvest each day. You get a whole pile of tubes. And clinical mastitis after treatment was checked weekly for the first, or daily for the first 7 days, and then once a week, weekly until 2 weeks pre-calving, and then daily checks resumed every day until 30 days of milk. Then at calving, all quarters were stripped 10 times in a pedal, a black pedal, so you could clearly see the teat sealant, and checked for present and absence. And presence means one fleck means present, and absence means that you see nothing, you see only milk. Yeah, so that's the difference. So there was no gradual quantity, it was just presence or absence. And again, the cow somatic cell count was collected automatically from the herd test. The circumstances that winter were really bad. That's what they told me, that winter was rainy, as it can be on the South Island in New Zealand. It was rainy and very cold. Yeah, and when cows are on pasture and you are palpating cows and it is very wet, the manure is so thin that this is how you end up after a while. And so the teat sealants were really tested. Yeah, so that's important to know. So at the end, the cows, 1,105 cows were enrolled, almost equal between each group, but 213 cows dropped off because they didn't have the right data, they were missing. Although checked for pregnancy, the 21 cows were not pregnant, or they were not present on the farm. They were culled, and so we lost 20% of the cows here. So 871 cows were randomly treated with the investigational product or the control product. Yeah, and here you see the farm characteristics. We selected 2 farms. And you see the somatic cell count, the bulk tank is very low, 110, 117. Milk production, 16 liters per day, which is, I think, 5,000 liters per lactation, typical for New Zealand. Average age, 4.8 years, and this was the number of cows that were in the herd. So big herds. And there were no differences, really. You see quite the same characteristics. The presence of teat sealant, you see that at the quarter level in the investigational product, 71.5% of teat sealant was present. In the control product, 67.1%, which is a difference of 4.4%. And at the cow level, there was a difference of more or less 5%. Yeah, you see here the number of animals, number of quarters, huge amount of quarters that you had to treat. So the investigational product increased the odds for teat seal presence with 26%. Yeah, and we said that the lower limit of the 95% confidence interval needed to be lower than 2%. and it was 1.6%. So that meant that both products in teat seal retention were non-inferior, but because the confidence interval did not overlap an odds ratio of 1, because the odds ratio was between 2 and 44%, so it did not go negative. The investigational product was superior. So that is the statistics of non-inferiority. That's how it works. So the decision was, the outcome was that the investigational product was superior. Mean this difference meant that it was non-inferior but also superior. Means that more teat sealant was present at calving in the investigational product than in the control product, more frequently. The cow somatic cell count is, you see the cows that were enrolled were very low somatic cell count, 22,024, which is very low. The number of cows that were treated with antibiotic, because they were having a high somatic cell count above the threshold in New Zealand, was 9 and 8. So you see that the far majority had low somatic cell count. And this was the days in milk when the herd test, the somatic cell count was tested. So that was not significantly different. The prediction of somatic cell count in the investigational product the mean was 84,000, and this was the 95% confidence interval. The control product predicted somatic cell count was 98,000, and this was the 95% confidence interval. Now, when was the product non-inferior? When this mean, 98,000, and this number, the upper limit of the investigational product, was not higher than, more than 10,000 higher than the 98,000. So if this number would have been higher than 108,000, it would have been inferior. But you see that it's 95,000, so it is within the 10,000 difference that we just said. And that means that the investigational product is non-inferior to the control product. Yeah, so it's a silly statistics, it's complicated, but this was the non-inferiority test. So even with the very low margin, you see that these products are almost the same, have almost the same effect in somatic cell count. Clinical mastitis, the difference was 0.74%. Again, here is the lower limit of the 95% confidence interval. It was below the 2% margin. So that meant that the investigational product was non-inferior to the control product. That means that for clinical mastitis products incidence, the products had the same effect. So the conclusion is that if you compare these two teat sealants under New Zealand circumstances, pasture, wet and cold cold environment, clinical mastitis, the investigational product was non-inferior. Somatic cell count, the investigational product was non-inferior. For teat seal retention, the investigational product was superior. But the clinical relevance of this superiority is low because there was no difference in udder health. So that is what this means. If you see more frequently teat sealant at calving, that does not translate into a better udder health. So that's why the clinical relevance of that is low. The study was published in journals Dairy Science in the beginning of this year. So if you want to read further details, you want to know further details, feel free to read it. And with that, I'm open for questions.
Thank you very much, Dr. Swinkels, for your talk. It was very interesting. Well, there's no time for a question, but Dr. Swinkels is here around in the Congress, so you can ask him after. For the next speaker, we have Ulises Jesús Bautista Pérez from Mexico, and we have the next topic is causes of sudden death in cattle, a practical way to differentiate them in the field. Thank you very much, Dr. Ulises.
Thank you. This one. Okay, yeah, thank you very much. So, good morning, everybody. I would like to thank first to the scientific committee for considering my paper. So now we are going to talk about some relative new science, at least in veterinary medicine. So this is the kind of animals we work with. This kind of, they are two students that are releasing the thesis, the final thesis, but this is the kind of animals that we work with. In this case, of course, it's complicated to release a necropsy where you can see maybe macro lesions in the liver, in the kidney, or something like that. So it's difficult, this part. Of course, in the early decomposition. So some of these animals that or cadavers or corpus that we saw are in late decomposition. So as introduction, here in Mexico is typical, I don't know, maybe more or less 50% of all the bovines that are here in Mexico are in total free grazing, where, of course, there is not not control overproduction, over-reproduction, no control overfeeding, and sometimes the farmers spend maybe weeks or months without supervision in the animals. So it is difficult to practice preventive medicine, and of course, there are no records. So in these cases, there are totally ignorance to cattle teeth, beards, and the purpose of this conference is, of course, deaths. As a clinical history, there is one idea, the main thing that the farmer said to you is that I found my cow dead in the morning. Because sometimes the people live not close to the farm or not close to the terrain where the bovines are. Some people have to spend maybe 3 hours, 4 hours until the system production. So of course there are no living there, and the next day they have the surprise or the bite surprise. So you have to consider in the anamnesis feeding, immunization protocols, vitamin, mineral, supplementation, livestock facilities as the other kind of practice in clinic or bovine, of course in live animals. So I would like to start just with infection diseases. I will talk about 2, clostridiosis and anthrax. Of course, I know that there are maybe other kind of infection diseases that cause the sudden mortality, but probably these 2 are the more prevalent here in Mexico and probably in other parts of the world. So I'm not going to describe these 2 diseases complete, just a practical way to differentiate. So what you can find in the inspection is typical that the vaccination over 6 months, animals with good body condition, several animals dead on different days, scavenging evidence, and increasing the composition rate. This increasing the composition rate is because the animal had bacteremia or septicemia, of course, of these two diseases. And the scavening for us is very important. I will show you next screens. So here you can see typical corpus of this kind of animals who die because these two infections. You can see here one bovine dead, but looks very well. The hair is shiny, good body condition, but the surprise was the same. No, just I found it dead in the morning. So you can see the good body condition. These are the other animal that, of course, is still alive. And you can see here calves. So it means that reproduction is well, of course, the nutrition is well. But finally, there are some dead animals. This is other kind of production, in this case, Bos indicus, but it's the same. You are going to see both, here is not very good, but it's good body condition. But finally, 1, 2, 3, 4 dead animals in this case with different rate decomposition. Other kind of causes of sudden death are the intoxications. In this case, I'm going to talk about nitrates, and cyanhydric acid intoxication. What you can find in the expansion, common in confined system. In fact, you can see it too in free grazing system, more common in the dry season, sorry. Of course, you have to consider changes in the diet, several animals dead, and this is important that in a few minutes, absence of scavening and decreases the composition rate. So the absence of scavening, in this case, is very important because these scavengers detected these substances, and of course, they are not eating these kind of corpuses. You can see here 3 bovines, in this case, were died maybe 26 in 30 minutes, all of them. So these pictures were taken by the farmers, and you can see here that there was not agony in these animals. In this case, these animals, we arrived there after 2 days after the event, so we couldn't work with these animals. So we decided to go just with the part of the terrain where they were grazing. So we took some samples and put them in tubes with sulfuric acid and chloroform, and these sticks with picric acid, where these samples or these sticks turn on red is positive to cyanhydric acid. Other kind of intoxication that is common here in Mexico is about snake bite intoxication. So here in Mexico, in this area, we have these main three snakes, as Botrox asper, Crotalus, and Micrurus in this case. Maybe this is the worst, because when these snakes bite the cow, could kill maybe one simmental or big bovine of 100 kilos, 1,000 kilos, sorry. What you can find in the expansion, common in grazing systems, following land clearing, usually one bovine death, abscess of scavening as the same idea as like the other ones, and graded degree decomposition in the bite side. So you can see here one bovine dead, a cow, and here you can see more decomposition grade in this downside, in the ventral side of the neck, in comparison of all the rest of the body. So you can think about this kind of intoxications. This is, of course, not an scavenger. It's just a little dog for the farmer. Another one cause of sudden death is ruminoreticular tympanies, and you know there are two, frothy blood and gas blood. You can find during the inspection that if you can see several dead animals, maybe it's probably the food, or one dead animal, probably esophageal obstruction. Of course, you have to consider the complete obstruction, because it's not complete, maybe partial obstruction. These bovines can, with other things, as a bovine leukosis or tuberculosis. That is common here in Mexico. Okay, I know that they are not cattle, they are sheep, but it's more or less the same. So you can see here the food is alfalfa, and in this case, the farmer wetted the alfalfa, of course, but because the ignorance sometimes of the people, and finally caused the death of part of the sheep. You can see here a typical lesion in the esophagus is the line tympanic or tympanic line. For us, it's very important because if you see in the necropsy, I know in early decomposition, not in late decomposition because you are not going to see it, but in the early decomposition, if you realize the necropsy and you see this tympanic line, the bovine died because the tympanies, because in post-mortem, you are not going to see this one. Other kind of is climatological causes, death by lightning and flood. Believe it or not, in Mexico, I think in other parts of the world it's the same, but in Mexico it's very common this kind of deaths by lightning. What you can find in the exhibition, extended limbs in early decomposition stage, but it's very important consider that it's not the same as rigor mortis. Rigor mortis is a stage, normal stage in the early decomposition. In this case, it's not exactly the same, but this is the typical, I don't know, position of this animal. It's common, see them under the tree. No apparent injuries upon necropsy if you release it. Lesions on the tree where the lightning struck, and multiple animals dead nearly simultaneously. Same decomposition timeline, and you can see now again scavenging. So you can see here two pictures. This one on your left side. There are 1, 2, 3, 4 dead animals. But the important here is that you can see here the lesions, the injury on the tree. This other picture, you can see an other tree, but in this case, the lightning destroyed complete this tree. So it's a very good sign, of course, for consider this kind of sudden death. Other one is flood. What you can find in this inspection, any type of production system near rivers during rainy seasons, several farms affected. It does not always rain in the area. Sometimes the rain is in the mountains or in other parts of the region, but finally arrive to the farms. Dead vegetation on fence, that is a very good sign for us, and sometimes, unfortunately, lost animals. So here are some pictures. In the first one is 2 days after this event. You can pass in this case. It's very dangerous. But these other 2 pictures are 5 days after the event. So now there is not too much flood. But you can see here on the fence this dead vegetation. that I saw that is a very good sign to consider this kind of sinister. In fact, you can see one guy here on a horse, and you can compare more or less the level, at least, of the water was. About trauma, there is many causes of trauma, of course. I would like just talk about two. Predator attack by canines, big cats, bears, and accidents. What you can find in the inspection, any type of production systems. In the case of predator attack, you can see blood stains in the pens. If the cows are the canines, many body wounds, pattern of attack not specialized or disordered. In the case of big cats, few body injuries, pattern on precise attack. And in the case of bears, big injuries, more in the dorsal region. So here you can see two calves from the same event by canines, in this case, dogs especially. And you can see here this disorder of the pattern of attack. You can see here injuries in the nose, in the ears, in this case on the limbs. So many injuries in this case. This one, sorry, this one was dead. The other one's alive. So they are no cattle, I know, but it's one sheep, one goat, but you can see the injuries in the neck because the puma, in this case, was. So it's relatively easy to determine this kind of death. And it's common here in Mexico. Puma and this region in the Yucatan Peninsula, in this part of Mexico, jaguar too. In the north of the country, you are going to see bears, but I don't have any pictures of bear. This is easy to recognize the kind of trauma, but look for the cow. So just one tree, all tree, because the air fell on the cow. So this is more or less easy to determine. As a summary, this is a table where I consider all these death causes. So it's just a resume, no? Where I consider time for the death to occur, number of dead animals, period between deaths, scavenging, it is present or not, and the other kind of evidence that you have, you can consider. As a conclusion, maximum attention on details, complete anamnesis, I said it before, as the other kind of practice in clinic, inspection of the corpse and the environment, caution with zoonotic disease, and comprehensive counseling with the farmer. Finally, I would like to thank first to my university, the main university here in Mexico. With the project, we realized a digital manual for determining the causes of death in bovines in late decomposition. And of course, I would like to thank all my dear collaborators to release this one. And finally, thank you.
Thank you very much, Dr. Ulises. So we have time for a break civic, but first we have 1 minute for 1 or 2 questions. Please, if you have anyone, you can pass on the center.
Okay, thank you very much again. In fact, I have the manual right now. If somebody want, this manual is of course in Spanish. You can translate it. I can share it to you maybe in the break. Thank you very much.
Hate to see you with some other, but are you happy? Hate to see you happy if I'm not the one driving. I'm so mature, I'm so mature, I'm so mature. Got me a therapist to tell me there's other men I don't want none. I just want you. If I can't have you, no, what should am I? I might kill my ass, not the best idea. His new girlfriend's next, how I get here. I might kill my ass, I still love him though. Rather be in jail than alone. I get this sense that it's a lost cause. I get the sense that you might really love her. It just can't be evidence. It takes this evidence. I touch a Russian with you, no murders, no crimes of passion. But damn, who is out of reach? Who is out the farm and sparkle with your perfect peach? Now I'm in the maze, man, plan, I'm impatient. Lying face down, got me singing over every. I'm so mature, I'm so mature, I'm so mature. Got me a therapist to tell me there's other men I don't want none. I just want you. If I can't have you, no one will. Am I? I might kill my ass, not the best idea. His new girlfriend's next, how I get here. I might kill my ass, I still love him though. Rather be in jail than alone. I did it out for love, I did it all on no drugs. I did it all this sober, I did it all for us. I did it all for love, I did it all this on no drugs. I did it all of this sober. Don't you know I did it all for us? I just killed my ex, not the best idea. Kill his girlfriend's next, how I get here. I just killed my ex, I still love him though. Rather be in hell than alone.
There's a little black spot on the sun today. It's the same old thing as yesterday.
Let's sway. White color lights up your face. Let's sway. Sway through the crowd to an empty space. If you say run, I'll run with you. If you say hide, we'll hide. Because my love for you will break my heart in two. Let's dance for fear your grace should fall. Let's dance for fear tonight is all. Let's sway. You could look into my eyes. Let's sway under the moonlight, the serious moonlight. If you say run, I run with you. If you say hide, we'll hide. Because my love for you will break my heart in two. If you should fall into my arms and tremble like a flower. Let's dance. Put on your red shoes and dance to blues. Let's dance to the song they're playing on the radio. Let's sway. You could look into my eyes. Let's sway under the moonlight, the serious moonlight. Let's dance. Let's dance. Let's sway. Let's sway. Let's dance. Let's dance. One knows what it's like to be the bedman, to be the sad man, the telling only lies. But my dreams, they aren't as empty. Not much love to me. I have hours, only lonely.
can show through. But my dreams, they are reminiscent teat. I have flowers. Love is conscience. That's never free. I'm so happy when I'm not with you. Then you saw me, caught you by surprise. A single tear to fall from your eye. I don't know why I ran away. I make you cry when I run away. You could have asked me why I broke your heart. Could have told me that you fell apart. But you walked past me like I wasn't there. I'm just pretending like you didn't care. I don't know why I run away. I make you cry when I run away. Take me back, 'cause I wanna stay. Save your tears for another. Save your tears for another day. Save your tears for another day. So make you think that I will always stay. Say some things that I should never say. Yeah, I broke your heart and someone did to mine. I don't know why I run away. Oh, girl, say I make you cry when I run away. Yeah, yeah. 'Cause take me back, 'cause I wanna stay. I realize that I'm much too late. Tears for another day. Save your tears for another day. Yeah, yeah, yeah. There's a feeling within, there's a feeling without. Try to clear to the left. There's a bug in my head, many bubbles all lost, but you never see the end of the road when you're traveling with me. And now, don't trip, it's over. And now, when the world comes in. Now there's love in the door, there's a hole in the roof. My position of destiny, vision of passing breath. In the back of the day, there's a war on our waste, but it's all my love is what you think. And now, don't trip, it's over. And now, when the world comes in. Now I'm walking again to the beat of a drum and counting the steps to the broken heart. Only shadows ahead, but we clear them from left. It's now the fading of the told me that you hated my friends. The only problem was with you and not them. And every time you told me my opinion was wrong, and tried to make me forget where I came from. I didn't wanna write a song, 'cause I didn't want anyone thinking I still care. I don't, but you still hit my phone up, and maybe I'll be moving on. I think it should be something I don't wanna hold back. Maybe you should know that Sounds like violence, break the silence, come crashing into my little.
Para que empiece. Einar, que si quieres.
Y así ya me camino.
Y para cambiarle estás
Sí, ese ya no,
familiarizado.
sí, sí.
Ok. Entonces nos haces otro micro.
5 minutos antes va a pasar el
Vibra,
amarillo.
ok.
Antes de que...
Hello, good morning. We are going to present Dr. Einar Vargas-Bello-Pérez. He's a veterinary zootechnician trained at UNAM with a master's degree from McGill University in Canada and a PhD from the University of Nottingham in England. He has been a full professor at the Pontifical Catholic University of Chile and the University of Copenhagen in Denmark and at the University of Reading in the United Kingdom. And now a full Professor B at the Autonomous University of Chihuahua. He has more than 165 publications in scientific journals and age index of 20. He has published 4 book chapters and has an extensive international collaboration network that includes more than 20 countries. He's part of the editorial board of different index journals, such as an Animal Tropical Animal Health and Production, Italian Journal of Animal Science, Livestock Science, and Health. His line of research focuses on ruminant production systems and the interface between nutrition and the quality of the final product of ruminants. Currently, he focuses on the application of artificial intelligence to improve production efficiency in milk production systems. So, we are very glad to have you here, Dr. Einar, and let's begin with his speak. He's going to talk us about smart nutrition improving milk fat quality of ruminants from Mexico. So,
Thank you very much, Mrs. Chairman. Good morning, everybody. My name is Einar, and this is a little bit of my momentums in life. And as she said, I've been in many places. I did my career in Chile, and from there I moved to Europe in Denmark, and then I went to United Kingdom. So I have seen many things, right? So today I will talk about, conceptually speaking, I will talk about smart nutrition, which means that we will configure a diet that will target something in the animal, right, to provoke a specific change. And in this case, I will be focused on milk fat. This is my research line, and as she said, I am within the interface of nutrition, ruminant nutrition, and product quality. So I have done in vitro cultures, I have worked with nutrigenomics, metabolomics, but today I will focus on feeding interventions to improve the lipid quality of milk, cheese, and ice cream. The approach is very simple. I always take in consideration animal nutrition, animal production, and public health, right? Because in this case, what I'm aiming to do is to reduce saturated fatty acids in the dairy products. So this is what we do. We work at the farm level, and from the farm level, we work on the lipid metabolism, and we want to increase bioactive compounds. And those that you know, probably conjugated linoleic acid is one of the most popular fatty acids that many people is targeting to increase in dairy products. And this is because it has been suggested or reported that this fatty acid improves cardiovascular health, right? So this is very important for the industry. This is very important for consumers. And the relevance for this topic is that this is the communication or a dialogue between animal nutrition, physiology, metabolism, until the consumers, right? Those will be the final users. And this crosstalk is very important, and not many of us see this dimension, right? So all these dietary interventions are meant to keep animal productivity, but also to increase those bioactive compounds. We have vaccinic acid, linoleic acid, and so on, right? So those are unsaturated fatty acids. So why do we talk about milk fat? Well, milk fat is very important for physical and chemical characteristics of dairy products. There is money around it, of course, because the farmer gets an extra quota when he sells the milk to the industry. And of course, it has a direct impact on human health. So this is, or that's why I found it very interesting. And this is also one of the components that is very easy to change with the diet, right? So, and also, of course, milk is a common food around the globe. We have done also studies on consumer expectations, and this is very important because our science is based on facts, right? So in one of the papers that we published some years ago, we asked people from UK, Denmark, and USA why they think or what do they expect in the milk fat, right? So we know that saturated fatty acids are the most popular ones. They have a little bit of knowledge, and between countries there are differences in terms of what they like, what they consume, right? So in another project that we did, we wanted to see if people like small ruminant dairy products, right? So what I'm saying is that this is very important because we can target consumer profiles, those that like this type of products, those that doesn't like, and from then I think that we can configure a diet in the farm that will fulfill those expectations for the consumers. So for the dairy industry, this is very, very interesting and important. So the first section of my talk will be about studies with sheep. And this one was done when I was working in Chile. This is a Mediterranean area. And if you see, I was working in the middle of the country. The country is a very large country. And I was located in a place where it's pretty much similar to Italy or Spain, right, in terms of environment. So we did a project next to a sheep farm where they had access to olive oil byproducts. And from that industry, we found that there were two byproducts, very interesting byproducts. One which is like a unrefined oil, and they call it lampante, which comes from lamp, because in the old times it used to be fuel to to make some light, right, in Spain. And then we also use olive cake, and the olive cake is the fibrous material that is the leftover when this oil is extracted from the olive. So we use both byproducts, and then we configure two different studies, one with a set of animals with use, lactating use, and what we did is to supplement the diet, and then we analyze the fatty acid composition in milk and cheese, and then the same for olive cake. So in both experiments, we have two different sets of animals, 9 in each of the experiments with 3 periods. They were arranged in a 3x3 Latin square design, and the diets were 3 diets. We always had one without this byproduct, and then the rest with a supplementation, about 3 to 4% inclusion of dry matter in the diet. So in the end, for example, the experiment with olive cake, we found out that we were able to reduce saturated fatty acids with 12, 14, and 16 carbons, which are in some cases bad for cardiovascular health or for the arteries, right? Some of them have been reported to increase problems such as thrombus or atherogenicity. In the case of the olive oil, we were able to increase oleic acid, which was one of the targets in the project. And overall, what we saw is that these byproducts could be used at least as a feed for small ruminants. So we published these studies, and it was a very nice experience. We just recently published with a colleague this paper about the use of chia seeds and pumpkin seeds on sheep diets. And we also found out that the use of these seeds were able to increase omega-3 and omega-6 fatty acids, which are also of benefit for human health. And we also were able to reduce palmitic acid content in milk of sheep. So if you see, we have been playing with different ingredients and see what is the best solution, right, to get the best quality in terms of milk fat. With other colleagues from Egypt, we recently published a paper using bacteriocins, which is a very strange name, but they are antimicrobial peptides. And we extracted them from, what's his name? Let me see, I don't remember. This is, well, we took them from cheese, and they were isolated from the manufacturing of cheese, right? So it was like a homemade bacteriocin. So we did 2 different sets of studies, one in vitro study just to check on ruminal fermentation kinetics, and also one with in vivo with animals, right, with lactating use. So we wanted to see if we could be able to use this bacteriocin instead of monensin, which is one of the additives that many people use also in dairy cattle. And we work with this bacteriocin, and we were able to increase milk production and feed efficiency. And also, it's not reported here, but we also improved the milk fatty acid composition in milk. Now I will go for studies with goats. And in this case, this is a study that we did in central Mexico. And in this case, what we did is to work with whole seeds of sunflower and linseed. And we supplemented goat diets. And these goats were fed with a control diet. They were arranged in a 3x3 Latin square design. And then what we did is to compare megalac. And probably most of you know what is megalac. Megalac is largely used in many dairy farms. And this is like a lipid supplement. It's made by hydrogenated fats. And many people in that area, they use it also for goats' diets, right? So we use that megalac as a control, and then we compare it with sunflower and linseed whole seeds. So overall, compared to this megalac, we found out that sunflower and linseed were able to improve milk fatty acid composition in milk towards a healthier profile for human consumption, and both didn't affect animal performance. So that was also a very nice experiment. And also in the same place, in the same university, what we did is to work with calcium soaps of palm, canola, and sunflower oils that were available in that area. In that case, we use a set of 9 Saanen goats. And we also work with a 3x3 Latin square design. We have a control diet, and then we have the supplemented diets. In this case, we use palm oil as a control because it was pretty much the same rationale as we use with the megalac. Megalac is mostly based on palm oil, so that's why in this case we use raw palm oil as a control. So that was a very nice experience. We were able to increase milk yield with sunflower, and canola oil was more digestible. Those were probably the overall findings from the project. Sunflower increased milk production, milk protein, and milk fat yields, which is very important for farmers because in this case, the goats were used to make cheese, and we need protein and fat for cheese production and yields, right? We found out that sunflower did not have deleterious effects on nutrient intake and digestibility. In another project that I did in Denmark, when I was working there, I had already a lot of knowledge on this lipid metabolism, on fatty acids, and in this case, what we did is to work with Danish Landrace breed. Those are very typical animals from Denmark. But in that case, what I did is to explore on metabolomics. So what we did, it was pretty much the same, but we also added the fact that we use 2 different ways of analyzing metabolome. Nuclear magnetic resonance and gas chromatography with mass, right? So we were able to improve milk fat from rapeseed and sunflower oils that were used to feed the goats. The rest of the milk composition was not affected, which is nice because we didn't provoke any negative effect on performance. And when we work with the chemical engineers, they told us that the NMR, which is a nuclear magnetic resonance analytics, they told us that the data was clearer compared to the grass chromatography because it works better for the goat's milk polarity. So that is a very chemical explanation, but all the results were based on NMR data. So overall, the animals that were fed with rapeseed oil, we found presence of metabolites related to alcohols and aliphatic compounds. And when we check on the literature, most of the literature is related to disorder at the rumen level. There are some papers that talk about acidosis, so clinical acidosis or not even subclinical acidosis with aliphatic compounds around blood. In this case, we don't have a clear answer how there was a transfer from the blood, from the feed, from the blood, from the rumen, until the secretion, which is the milk. But this is probably one of the first reports on these elements. Sunflower oil increased some metabolites related to protein metabolism, and that was also a very interesting finding. We published 2 papers, one based on foodomics and the other one based on in vitro experiments. So in the end, we have a whole explanation, at least from the metabolism, right? From the metabolism point of view. We recently, and this was just published last week, we used calcium soaps from garlic and willow extracts, and we use them to feed goats. So we check on nematode loads. It was like an antiparasitic supplement. We analyze nutrient intake and digestibility, and we also check on fermentation kinetics at the rumen level. So we use both extracts, and we were able to increase the digestibility of both dry matter and organic matter. We were able to reduce fecal egg counts of some parasites, such as Emuncus and Trichostrongylus. So that was also an interesting study. So now I will go for studies that we have done in cows. And in this case, when I was working in Chile there is a cheese. Is one of the most typical cheeses that are sold in that country, and the name is chanco cheese. So what we did is to have 9 Holstein cows. The animals were arranged in a 3x3 Latin square design, and the animals receive a control diet, and then they receive a diet with soybean oil and also with hydrogenated oil, right? So we wanted to see if we were able to increase unsaturated fatty acids with those animals fed on soybean oil, and to increase those saturated in those animals fed on hydrogenated vegetable oil. The interesting thing here is that we also have a sensory analysis on the cheeses that we produce, and we check on 16 attributes related to appearance, odor, flavor, and texture. So this was done probably in 2014, and that was one of the first experiments in Latin America that were using this concept of from farm to fork, right? So we had the whole explanation on lipid metabolism from the animals, and we also have an explanation from the sensory analysis and also physical and chemical analysis from those cheeses. Those are the experimental cheeses. And of course, the animals that were fed with soybean oil, they showed an increase in vaccinic acid and monounsaturated fatty acids. And we were able to decrease saturated fatty acids and thrombogenic index, right? Which is a calculation that more or less gives orientation on how bad is that product and if it can provoke a thrombo when a human consumes this type of cheeses. We didn't find negative, there was no negative effects on milk and cheese chemical composition, which is good for the industry. And we didn't find differences between treatments in terms of sensory characteristics, which is also good, right? Because we have a cheese that has a bioactivity and it tastes well and it looks well, right? That was one of the objectives that we were looking for. In the same project that we had, we also used fish oil. The fish oil was based on salmon oil because by the time I was working in Chile, and Chile is one of the main producers of salmon oil, so we had plenty of that type of material. And then we made a blend between fish oil and hydrogenated palm oil. So we had pretty much the same arrangement. We work with a 3x3 Latin square design, and we check on these sensory characteristics and physical and chemical characteristics. If you see in the picture, I have a student that is taking pictures from the surface and then from the core of the cheeses. And this is very interesting because we correlated those results with the ones that the judges reported on the sensory analysis and the ones that we got from the pictures, right? So in both cases, what we found is that the cheeses that were made from this fish oil treatment had an intense yellowness. And in this case, in the Chilean market, this is what the Chilean consumers are looking for, a bright yellow cheese. So in this case, it was a very well-rounded project because we could be able to increase those bioactive compounds, and we were also able to improve the physical appearance of that product, right? So that was one very nice experience. By then, I was curious about doing another type of dairy product, and in this case, we did ice cream. We work with 15 animals, and in this case, the animals receive also different types of oils. In this case, what we had was soybean oil and fish oil, two different oils that, for example, soybean was able to increase unsaturated fatty acids, and fish oil was able to increase large chain unsaturated fatty acids, right? So we did the ice creams, and then we also analyzed sensory characteristics. Those are the experimental ice creams that we were using. Of course, the judges were not able to check on the name of the treatments, but they were pretty much like the ones that you can find in McDonald's or Burger King. So in the end, the experience was very interesting because we were able to improve the milk fat, but at the same time, there was a very high transference of long-chain fatty acids from those ice creams made from fish oil. And the problem was that they melted very fast, right? So when you have an ice cream, you always like to feel these crystals, ice crystals, right? But as consumer, when you have a melted ice cream, it's not very nice. So in this case, probably this was a little bit of a failure. And this is probably one of the examples that I told you in the beginning. We need to improve our communication with the food science professionals, because probably we can be able to improve fatty acids or diets at the farm level, but we don't know what will happen when we go for technology and manufacturing of dairy products. So this is a very nice and interesting example that we found. In another project that we did in southern Chile, we were working with brassicas, which is a special forages like a turnip, and the other one is called rape, right? Those are the pictures. So they are like tubers that are found in that area. And in this case, what we had is the control diet without turnip or tubers, and then diets with that forages. We manufacture the chanco cheese, the Chilean cheese, but in this case, in the attributes that were related to sensory characteristics, we added two different ones: spiciness before swallowing and after swallowing. Why spiciness? Because we already knew that these brassicas, they transfer glucosinolates, and glucosinolates are related to spiciness and this type of flavors. So that's why we added these compounds, because we were considering that probably we were able to use the forages, but probably those farmers that normally produce cheese, they will have some problems with the taste of that cheeses, right? So in the end, we were able to check on the sensory characteristics on both turnip and rape diets. They produce cheese with greater intensities of flavor, aroma, spiciness, before and after swallowing, bitterness and acidity. So if you see here, we were able to use the forage, but if a farmer wants to manufacture cheese, then we will have problems because there is this type of transference of glucosinolates to the final product. And at least in Chile, it's not like Mexico. The Chileans don't like spiciness. So this is probably another failure that we had. We were able to improve other things that we were looking in the project. But in terms of the sensory characteristics, it was not nice for that market, right? For the Chilean market. Today, what we're doing in central Mexico is work with insects as a sustainable feed alternative, alternative to the use of soybean meal and fish oil meal in some cases. We have done some trials using larvae from black soldier fly, from yellow mealworm, and from aquatic backswimmer insect, right? Those are the pictures from that insects. And at the moment, we have done a whole screening of in vitro studies, and we have been checking on ruminal fermentation characteristics. But we have also checked on different characteristics from dairy animals and also from lambs meant to be used for meat production, right? These pictures were taken in 2020, 21, when we had this pandemic problem. And when I was visiting my colleagues in Mexico, the lambs that were fed with tenebrio, that insect, they show that compared to those animals that were fed with soybean meal or fish meal, they had a smaller brain. So that was a very interesting thing. And of course, if you see, I already had like 15 years of experience working with lipids and oils and all these things. But for the first time, I saw something different from the dairy products. So we dig more into this thing. At the moment, we don't have the right answer, but we publish a scientific reflection trying to see if we were able to use supplementation of omega-3 or omega-6 fatty acids during the last trimester of gestation in ruminants. At least in this paper, we use the sheep as a model. And those fatty acids are pretty much related to neurological development, cognitive development, and behavior. So we pretty much hypothesize that if we use, for example, linseed or any other oil seeds enriched with omega-3 fatty acids, probably they will lead to a very nice result in terms of cognitive development and behavior. So that means that the behavior of the animals will be probably better for the system, right? Probably they will be able to adapt better to the feeding management, to the milking management, and so on. So this is just an idea that we just published. So just to start to wrap up the messages, I think that it is very important to establish this communication between veterinarians, animal scientists, agronomists, and the food science sector. We found out that the chemistry of dietary lipids works, it's not like a black box. It always has different results, and most of them can provoke good changes in terms of animal performance. In terms of product quality, they can also provoke unwanted changes, and we need to work on that. And we have done many trials with farmers, and the problem that we have encountered is that when we do this type of on-farm studies, we cannot prevent these adequate designs or things like that, right? So we always have to deal with them and see how can we adapt to their daily routines. And this is something that we need to improve, and probably most of you that do experiments is probably a really big pain, right? So those are the challenges in terms of the science that we're doing. What we see in the future is that we can improve the lipid quality from dairy products from ruminants. That's definitely doable. This research approach could be useful for niche markets in different countries. The dairy industry, if they identify niche markets, then these products could be sold. and not very cheap. They could be sold at a very nice price. And there might be other players in the dairy industry that might be interested. And if you see, we don't work with the skim milk. We don't remove the fat. What we do is to get a whole milk, either as a liquid milk or as a dairy product. And what we work is on the nature of the chemistry of the lipids within that whole milk, right? So this is not like just removing the milk like everybody do in the dairy industry, right? They centrifuge and then they extract and then they can configure whole milk, semi-skim milk, and skim milk, right? So in this case, we just work on the chemistry of that whole milk. I would like to thank all my collaborations. This is just an example of people that I have in Mexico. We work with different universities. And around the globe, we work with more than 15 countries. This is not just an individual effort, it's an effort from many, many people. And also I thank all the people that is involved in the farms, the animal students, universities, research centers. So now, thank you very much, and I will be able to answer your questions.
Thank you very much. If you have any questions, this is time for. It was a very, very nice speak. Thank you very much. So, please come
here. In Europe, insect story is now gaining popularity. So insects are considered first of all for feeding the chicken, especially broiler chicken and other poultry. And also there are some trials on feeding the piglets or pigs. And there are more, how to say, refrains considering human food as a source of protein fat. But to my knowledge, this is a relatively expensive feedstuff. I mean, the insects of various species you showed. So my question is it practical to use such relatively expensive feedstuff for the ruminants?
Yeah, that's a very interesting question. We have discussed this with our research teams, and of course, I will not sell the idea that we can use it on a large-scale production. First, we make basic science, so we just want to prove some other things rather than just trying to sell the idea that you can save the world with the insect use, right? When I was in Europe, I know that the European community has a very big problem on biosecurity because we don't know what will happen when we go for a very large-scale production, especially for the black soldier fly larvae and other type of growth developments from that insect. But in this case, in Mexico, first of all, we don't have a right regulation. There is no regulation about the use of insects. In Europe, you're starting to have problems using this type of feedstuff. That's one thing. The second thing is that probably in areas, and especially in Mexico, it's a very large country, areas where you can find not black soldier fly, but other type of insects that are around your region, then probably you can use it to a little, I don't know, like to increase a little bit of the protein that you cannot find with the grasses that you use to feed the animals in extensive systems. So that will be one of the bets, at least in the Mexican situation. But you're right, it will be very difficult to convince someone to go for a large-scale production of this. In Scandinavia, when I was working in Denmark, if you go to the supermarkets, you will find these insects, but only for human consumption. And then they sell you these small bags, very expensive, and you can find worms, right? But yes, that's what I know. Thank you.
Thank you. Any other question? No? Oh, well, thank you very much. It was really a very nice and interesting talk. So if we don't have something else to ask, let's give him an applause.
Thank you.
And let me give you your gift for being here. We're going to have some minutes free for start with the next conference. So I would like to know if is here Katie Waine from Canada? Yes. So we're going to start in some minutes. If you Hello, good morning again. Thank you for the new ones that are here. We are going to start with our next conference that is named Extensive and Served Necrotic Tongue Lesions in Canadian Feedlot Cattle. And we have here, it's an honor to have you from Canada, Katie Waine and Alberta Vivhill. No, I'm on Alberta, what's your name? Alberta, yes? Megan. Megan, it was difficult to read. Yes, Megan Brockhart, I'm so sorry. So, we have you, so you're free to start. We have 15 minutes.
Thank you. Good morning, everybody, and thank you for joining us. So my name's Katie Waine. I'm the veterinary pathologist that worked on this case with some of my colleagues at the University of Calgary. And with me today is Dr. Megan Brockhart, who was the clinician working at the feedlot. So we're going to present to you a case of severe necrotic tongue lesions in cattle in a feedlot in Alberta, Canada. And I'm going to pass over to Dr. Brockhart for her to go through the clinical presentation first.
Hello. So I was the veterinarian that got called out to this case. And on the 23rd of July in 2023, there were 3 feedlot cattle at a heifer-specific yard that were identified with severe tongue lesions. This yard was at about half capacity with approximately 12,000 head at the start of this outbreak. Within 7 days, so the first week, 77 cases had been identified in about 30 different pens at the unit. Over the following 6 months, we had over 1,200 cattle affected, so approximately 10 to 12% of the yard. In the summer of 2022, there was a previous cases were identified at a feedlot owned by the same company. They were about 5 kilometers away. Significantly less cattle, but we still did an extensive testing, including field necropsies, virology, bacteriology, and a lot of feed testing. At that time, there was moldy corn that they had been feeding, and the stopping of it appeared to correlate with case reduction. We had heard from colleagues in the area that over the last 10 years, there had been some cases with similar numbers, actually, and they had done extensive workups, but no definitive cause was found. One of the things that we used pretty extensively in the initial stages of this workup, both in 2022 and then again in 23, was a Nebraska case report from 2001 that showed similar lesions in feedlot cattle in Nebraska. So we knew this wasn't a new problem, but at this point, no cause had been identified. So the pen checkers and the foreman, the feed truck drivers were all critical in identifying these affected animals. It became a little bit of a team sport to get them identified and quickly managed. These cattle would be identified in the pen due to drooling, bleeding from the mouth, protruding tongues, or unusual behavior at the feed bunk or at the waters. They also had this thing where they would tongue twirl, which is kind of just like playing with their tongue, but to the point of excessiveness. And so the guys at the yard and the women, they would pull these animals, and this middle photo was taken by a feed truck driver. So the primary lesion found in these animals was affecting the rostral tongue, and it could range from a small defect in the surface mucosa to a huge loss of tissue. We had animals that were missing a third of their tongue. So all of these animals were brought into the chute and examined there. The tongue lesions were scored using a 1 through 6 point scale to describe the severity. Number 1 was a single lesion smaller than a loonie, which is the Canadian dollar coin, and then 5 or 6 was large portions of the tongue was missing or massive hemorrhage. The animal ID was recorded alongside a photo of the lesion location, whether it was right, left, or central, and presence of any neurological signs, and then the treatment that was given. The treatment that we came up with was something we used in 2022, which included ceftiofur antibiotics for 3 days, removal from their ration in their home pen, and placed on a low concentrate ration with hay in a hospital pen. If they were severely affected, Canada has options for emergency slaughters, and so we did have animals that were emergency slaughtered if they were missing large volumes of their tongue or were excessively bleeding. So in the early observations, we saw that it was primarily the heavy cattle that were affected. The average days on feed was 266, and the average weight was about 600 kilograms. They were on a high ration with MGA. We said before, this was a heifer-specific yard, so they were all heifers. In 2022, it was a mix of sexes, and the hide color was predominantly black. This schematic shows where the cases were located from the 23rd of July to about the 15th of September. And the X's on the squares are where they actually didn't have cattle. So I said this yard was at about half capacity. So they had either left the premise or they were getting ready to fill some of these yards or doing maintenance on the pens. And so there was almost every pen had at least one. And some had multiple. So at this point, our differential diagnosis was trauma to the tongue from the waters, the feeders, fencing, or the equipment, feed toxicity or a water toxicity, infectious, viral, or bacterial. So we spent a lot of time in the environment going through what could cause this. The water was sourced from an irrigation district line. We looked for stray voltage, for sharp edges in the waters, different types of waters. We looked for sharp nails or loose wires. A magnet was run through the limestone bin, the corn bin. The truck magnet was checked multiple times. There was nothing sharp identified. They didn't have access to any free equipment anywhere. In the management, in August, we decided to split one of the pens because it was quite hot to put less stress on heat abatement resources, and that really did no effect. And then this company did aerial spraying for flies, and they had done it June 2nd, but elected not to repeat it, but at their other yards they did with no adverse effects. So we spent a lot of time looking at the feed, and we've come back to this a couple of times, but we looked at the DDGs starting in May and June. They were new, and they would rotate around multiple piles on the premise. When it started, they were using the north pile. On the 25th of August, they switched to the south pile of dried distiller's grain. And in the high ration, it would be a 21% inclusion. In the low ration, it was 7%. We looked at the roller for corn when it was regrooved and cleaned and how. We chose to bring silage from another yard owned by the same company for the month of August, but driving it back and forth was a little bit extensive, so by September, they decided to stop it. The other thing at this point, we were just getting frustrated. We really hadn't found anything or what was correlating and thought, well, maybe it's multifactorial. It's hot, it's the summertime. The cases did drop after a rain in mid-August, but really no case reduction with that temperature change where it's cool evenings. We were doing export training at this point, or exporting, and so the large animals were running through the chute, and so we thought, well, maybe there's some handling stress involved with that because we would see a few more cases after they had been run through the chute, but still no obvious correlations. We did a lot of on-farm diagnostics, and while we did circle back to the feed because this was probably our primary diagnostic, everything was really unremarkable and pretty frustrating to get readouts of we can't find anything wrong with these animals. And then on that note, I'm going to turn it over to Dr. Waine because they helped us quite a bit at Calgary, and so she's going to go into the clinical pathology.
Okay, so we actually started by receiving a couple of heads to examine, which was really useful because it meant we could have a look in the mouth, we could examine the brain, the nerves, and the blood vessels of the head. But consistently, the main finding was this really severe necrotizing lesion affecting the rostral 2/3 of the tongue. And on histopathology, we've got H&E stain on the left, which shows, as we would expect, a loss of tissue, a lot of inflammation, and some necrosis and inflammation of the underlying muscle. This is a gram stain on the right-hand side, showing lots of mixed pink and purple bacteria, and we considered these to be commensals of the mouth and/or secondary invaders. So because we knew that there'd been some of these similar outbreaks happened before and nobody had been able to find the answer, we knew quite early on that we weren't going to find the answer sort of straight away, and one of the first things we did was a literature review to see if we could find anything that might suggest any clues, either in cattle, other veterinary species, or in people. There were similar lesions that we found in people and also in companion animals, but nothing that would really account for this huge number of animals affected in quite a short period of time. So our differential diagnoses at this stage, we were trying to determine, do we think this is a primary tongue lesion? Have we got trauma to the tongue? Could there be trauma to the tongue secondary to a behavioral issue? Could this be an odd presentation of Histophilus? Maybe a clostridial disease? Do we have treponeme involvement? And then we were thinking along the lines of the neurological. Could we have a local tongue neurological issue? So is there something that's causing a loss of sensation and subsequent damage by the teeth? Could the tongue be tingling? Or have we actually got a central nervous system lesion? And we didn't think there was a sort of a really severe neurological involvement because these cattle could still feel and they could still move their tongue. So we considered that to be fairly unlikely, but a local sort of sensational tingling we still consider to be possible. So we did go ahead and we did quite a lot of diagnostic testing. We ruled out a clostridial cause with fluorescent antibody testing and immunohistochemistry. We did salmonella and Histophilus immunohistochemistry on tongue tissue, as well as listeria and bovine herpes virus 1, IHC in the brain. We did a lot of virology testing. We also sent tissues for the mineral panel analysis and also for toxicology. We sent off somewhere between about 10 and 15 samples of feed for mycotoxin, ergot toxin, and toxin screen testing. All of that initially came back unremarkable, and we spent a lot of time talking to colleagues in Canada and further afield to see if anybody had seen anything like this. I was really quite interested to try and find out how these lesions were starting, but getting hold of more acutely affected animals was actually quite difficult. The few that we did find, we found some of these lesions that appeared to be wedge-shaped, extending up to the surface, with some hemorrhage and occasionally some necrosis of the muscle underneath, or some very well delineated lesions. And at this point, and still, we're trying to decide, is this something that's starting from the inside and going outwards, or is it something that's starting from the outside and going in? So in about September, having had all this feed testing that had come back negative, the clinicians on the farm realized that there were these clumps, really hard clumps in the dried distiller's grains, that were, some of which were being broken up in the mixing wagon, and some of which were still hard and entire in the feed troughs. So at that stage, whereas all the previous samples had kind of been mixed together for the toxin screening, we actually sent off 6 of these different samples for mycotoxin and ergot toxin testing, and some of the really hard clumps, especially those that weren't being broken up, came back with really high levels of ergot toxin in these really high clumps. And we noticed, so the cattle that were affected were the heavier cattle. They had a higher percentage of distiller's grains in their diet. And we also realized that the younger cattle would tend to leave these clumps. They would be left over in the feed troughs. But the older cattle, there weren't as many of them left over, raising the question of whether they could be eating them. So when we also when we had enough cases to be looking at the numbers, we could see that we had also had sort of peaks and troughs of cases over the months that this was happening. And we also realized that the south pile of distiller's grains had been introduced on the 25th of August, and then we had this huge rise in cases again on the 27th. And it was the south pile of distiller's grains that also had a huge number of these really hard clumps of mold in the feed. So the current situation, we've had over 1,200 cattle affected with these tongue lesions. We have found really high levels of ergot toxin in these clumps, in the dried distiller's grains piles, and the introduction of this feed appears to correlate with an increase in cases. All the affected feed has been discarded, and so far we haven't had any new cases since the end of January. So we're wondering at this stage whether this could be a case of local ergot toxicity, which has been reported in other species, although in different circumstances. We also acknowledge that perhaps this has nothing to do with the ergot toxins, and there could be something else that we're completely missing. So we really hope that some of you have some ideas to share with us today, because we certainly don't know that we have the answer for this. But if you do have a similar presentation or similar problems, we would definitely suggest looking at the feed first to see if you find anything similar. So we have a big list of acknowledgements, but I would like to start by acknowledging Dr. Barry Blakely. Barry was a professor at the Western College of Veterinary Medicine in Saskatoon, where he was a toxicologist. He was involved with this project, and he was really helpful and gave us a lot of ideas. He very sadly passed away very suddenly a couple of months ago, so we'd like to acknowledge the contribution that he made to this project, but also it's a really huge loss to the veterinary profession in Canada and certainly worldwide as well. We'd also like to thank the owners of the cattle and the team at the feedlot. This was really tough. They were seeing a huge number of these cases every day, pulling these cattle out, and they were really collecting a lot of information to help us work through the case. Thank you also to the vets, techs, and staff at Alberta Beef Health Solutions, to the team at the Diagnostic Services Unit at the University of Calgary, to our colleagues that helped us at Prairie Diagnostic Services, Animal Health Lab Guelph, the University of California Davis Animal Health Lab, and many others. The investigations were partly funded by the producer and partly by the Veterinary Outbreak Investigation Service from the University of Calgary. And a huge thank you to all the colleagues in Canada and further afield that have shared their thoughts and opinions on this case. So thank you very much for listening. We welcome any questions, thoughts, and especially if there's any experience of similar cases. Thank you.
Thank you for this very nice conference, and it's a really big challenge. So if you have any questions, we have enough time. Yes, please.
Thank you very much for sharing your experience. I have a lot of experience with ergotism in swine. And normally, ergotism in cattle comes along with tail or ear necrosis, or even with fescue fever and coronary inflammation. Did you notice such symptoms? And my second question is, there is not an ergot toxin. To get a lab analysis, it's an amount of 12. Did you do a specific analysis? Which kind of the ergot toxin was it?
So your first question, we discussed this with the toxicologist because we didn't see any of the other signs of ergot toxicity, which is one of the first reasons why we're like, could this be real or could this be just a complete red herring? And so one of the theories that the toxicologist suggested was because these clumps were so hard, so some of them, the clumpy material that you saw, some of it got broken up and dispersed through the rest of the feed, and when we tested that, the ergot levels were within normal limits. Some of them didn't get broken up, so we were wondering whether if these cattle were picking up this stuff, playing with it in their mouths, whether it would be possible to have a very localized effect, rather than by the time anything gets through to the rumen and it's mixed in with the other stuff, there's then not enough of it to cause a systemic problem. So that was one of the possibilities that we were thinking. But again, we're really, to find it was really interesting, but we certainly don't necessarily think that we've found the answer here. So it's a possibility, and we've been looking at some of the literature around the tongue necrosis in other animals, but we are very aware that it might not actually be anything to do with these tongues, and we could be looking at something else altogether. And with regards to the testing, we were trying to simplify it for the presentation. We did send it off to one of the labs. We have all the results with all the breakdowns of the toxins. The bar testing that we had, did just call it ergot toxins? With it off the scale?
They split it into two types.
Okay.
Yeah, there were two main types. There were two main types of ergots that they On the results from Alltech is where we sent it off to, and they did 2 different types of ergot, and I apologize, I don't have their names in front of me, and I'd have to look at them and read them, but we do have them in our email, we just wanted to simplify it for everything. And both of them were off the charts.
And third question coming up, a toxin is very affecting superficial skin is T2 toxin coming from barley. And it makes a very burning sensation when you eat it. Though it was tested for T2 toxin too?
So the DDGs were supposed to be corn, and they were actually not feeding any barley at this time. So we didn't actually think that that was going to be an issue. However, we ended up testing some of the distillers' grains to see if they were actually contained gluten, and they did. So they did not get corn distillers, or we don't think that they did. It's kind of an unregulated drop when they drop them at these feedlots. So we think that they may have gotten barley, which would maybe correlate with some of the responses I got from the foreman, where early on in September, the cattle that were affected were a little bit crazier than normal, like a little bit high-strung, and just it was the statement was made, don't go in that pen unless you absolutely have to, or on a horse, or someone can pull you out. So they were a little bit crazy. So thank you.
So, thank you very much to Katie and Megan. I know there are some other questions, but we are on time. So is here Carolina Ferenc. Thank you very much. If you want to. And please check it if you are familiar to it, or if not, I'm going to explain it.
No, it's just like.
Yes. Very much. So we're going to have the next conference. That is the number of white blood cells in European bison increases in teat, quirk, or the rule, preliminary data. So she's coming from Poland, and thank you very much. We are going to have 15 minutes. So.
Okay. Okay, so thank you for coming for this session. I know that the topic of European Bison, even for Buiatrics Congress, is very specific, narrow specialization. So, but for Poland, and of course I'm coming from Poland, it's a very important topic because we are responsible for the preservation, for conservation of this species. So even if we are very important producer of milk in Europe, we are on the 5th place, as I checked recently. Also in the world, we are good producers of the milk. Today I decided to tell you about European bison and our research in this area. So first of all, maybe it will be better to explain you what is the European bison comparable to American bison, because American bison population is much larger, and it's around 500,000 of individuals, but European bison population is much smaller. It's just 7,000 individuals. Of course, except that they are in different continents, and maybe for the first look they are similar. We have several differences. Okay, differences, they are not in size. As you can see, the size is very similar. But if we compare the anatomy, we see that we have some differences in the number of the ribs, for example. So in a European bison, we have 14 ribs. In American bison, it's 15. Also, European bisons are taller, they have longer legs. They are less hairy, except of the end of the tail. They also have different shape of the horns, and these horns are more similar to the primitive breeds of the cattle than in American bison. And okay, they are run smaller, but they can jump higher. Okay, so it was just short introduction to show you how this animals looks. Of course, in the Europe, it's the biggest wild mammal where you can meet. Yeah. And in the past, this population was much larger, but during the First War, it was problem with European bison population. It was destroyed, and we rebuilt this population. So now in Poland, it's the 1/4 of all population live in Poland, and we are responsible for this population. We are responsible for their environment to keep this species to next generation. The most of these animals, as a free living animals, they live in the Białowieża forest. So you as a Buiatrics, if you will visit Poland, for sure you should visit Białowieża forest to see this wonderful animals in their natural environment. So it's our national park. If we talk about the health monitoring, we are doing this health monitoring, and as you can see, some episodic status of European bison is quite good known. So we know that, for example, Schmallenberg virus, it's these animals, around 70% of animals, they are seropositive. Also, bovine respiratory virus type 3, it's common. If we talk about some bacterial diseases, Leptospira or Mycobacterium are common. Parasites, for sure, Trichostrongylus family. So, for example, Pneumonesia, Nematodirus, but also flux. So, Fasciola hepatica also. So this status, if we consider some infection diseases, parasites, it's good known and monitored. But we found that to evaluate the health of these animals, when we check just simple blood test, we are compare the range of our results from bison to cow range, which is of course good known. But we start to think that maybe it's different, and maybe we should start to build some data to find that the European bison range for blood samples, for blood parameters, are different than for cow. And it is good known for American bison, that you can see that some of the parameters. Some of the parameters are close, but not the same as for cow. So we have ranges as a green for cow, yellow as an American bison, and you can see that sometimes they are close but not different, and sometimes they are completely wider than for cow. But we don't have this type of the data for European bison, so that's why we start to build this database. Some of the parameters, even for American bison, are still don't know. So, for example, iron concentration in the blood, some, for example, lactate as a biochemical parameter of the blood, they are still don't know. And if we want to compare, we are rather use cow parameter. Now, as you see, very fresh manuscript. It was published during our flight to the Cancun, but also from the team from Poland, which in this publication they found for sure that we have some differences in the blood parameters dependent of the sex of the animal. And also they found some ranges, so now our results we can also compare with these results. Okay, so as I told you, we want to find what is the optimal range of blood parameters to better monitor the health status of the European bison. What we use, so of course the number of the bison which we can immobilize to take the blood sample, it's limited. We are doing it periodically, but in short time it was quite hard to find more samples. But we take 7 samples from 2 different areas. So as you see, first it was the Białowieża forest, so the place where we have the most population of the European bison, and the second one, it was Kruklanki forest, so it was different population which is not mixed with this population from Białowieża forest. And we take the samples in 2 different seasons. So we take samples during the summer and also during the autumn. And we analyze these samples using simple hematology analyzer and also using gasometer. And gasometer parameter were done in place. So for this type of analysis, it's very important that it was done immediately after taking the blood sample. So as I told you, first we should immobilize our European bison, so we used Palmer gun, and it was atropine mixed with xylazine, and after put them down, we did the clinical examination, full clinical examination. We monitor basic parameters during this sedation, and animals after this clinical exam and taking the samples, I think after 15 minutes they were wake up. It was just short immobilization of these animals to take samples and perform clinical examination. So animals were clinically healthy. We not found any health problems during this clinical exam. But what we found, maybe what we not found first. Red blood cells number, as you see, we have, we differentiate our samples for this which were taken during the summer and during the autumn, and we compare these ranges with cow and with these ranges, reference ranges for American bison. And we found that if we take to consider red blood cells, hemoglobin or hematocrit, this or also platelets, these ranges are, cow ranges are good to use to check the healthy of animals. But if we consider different parameters, as for example mean corpuscular volume or mean corpuscular hemoglobin or mean corpuscular hemoglobin concentration, you see that the data from the cow are completely not fit to our results. So here maybe closer, better it will be used the data and ranges which were estimated for American bison. Okay. The next, what we found, that the number of white blood cells are completely different between the seasons. So during the summer, the number of the white blood cells were similar to this for cow or for American bison. But during the autumn, it was the middle of the autumn, so it was dramatically drop of the temperature in this time. We found big increase of white blood cells, and because of it, of course, lymphocytes and neutrophils population. So we hypothesized that maybe this drop of the temperature can impact of these results. Next, basophils and eosinophils. As you can see, the ranges were similar to this for cow. pH of the, if we take to consider gasometry results, we see that pH of the blood was maybe different than for this for cow. If we want to compare PCO2 or PO2, we cannot compare it with data from cow or American bison. They are not exist, or I cannot find them. Also the ions. Ions were in range for sodium, for chlorine, for potassium, but not for calcium. Calcium was much lower than these ranges, which are estimated for the cow. Glucose level also were different, especially in the summer, was much higher on the atom also than for cow. These ranges for American bison, maybe they were better, but still during the summer we found high level of the glucose in our sample. Also the same it was with lactates. Okay, so here is the summary of our results. They are just preliminary results. We want to build a bigger base, but for now we see that some of the parameters which are to this time we use to estimate the health of European bison using cow parameters, cow ranges are okay. So red blood cells or hematocrit, but some of them, they are completely not fit. Some of the parameter is better to use American bison parameters, and some of them, they should be investigated in the future. So, yeah, they are my conclusions. So future investigation maybe for the next Buiatrics Congress. And thank you so much for your attention.
Thank you very much, Carolina. And if we have some minutes for if you have any question. Thank you very much.
I wanted to ask you the PCO2 and PO2 which you calculated.
Yeah.
Was that arterial blood or venous blood?
It was, okay, I didn't tell you, but it was venous blood.
It was venous blood?
Yeah.
Okay. You know, xylazine itself is known to cause some drop in oxygen in ruminants. I don't know about bisons, I've never worked with them, but in sheep and in cattle, it also causes a reduction in PaO2. I'm not saying PVO2, but arterial blood oxygen goes from 90 to 30 sometime within a minute. So I'm just wondering if, because you use it orphan and xylazine, if xylazine has some effect.
Yeah. And we completely know about it, and we also consider it, because we are not possible to take the blood from the European bison without immobilization. And our protocol of the immobilization is also always the same. So for us, it's still benefit to estimate this range for PCO2 after using this xylazine and after immobilization, because we don't have any different option to take this sample. So for cow, for sure, it's not always needed. We are not using the sedation to take the sample, but for European bison, we don't have another way.
I'm not saying that the work is important.
Yeah.
I'm not underscoring the value of that. But go ahead.
Okay, thank you very much. We have time for our last question, please. Yes.
Just a very short addition. We have also data regarding the time. So we have blood gasometry results immediately after immobilizing the animal and 15 or 30 minutes after. The changes are really minimum, 5–10% maximum concerning the data.
Thank you very much. So we're glad to have you here. Very nice conference. Thank you. And we are going to start with the next one. Okay. So your plan? Yes, is that well said? So almost. But we're going to have the next conference. It's Plum Petterman. Okay. So if you want, you can have this microphone, or do you prefer that one? Okay. And there's the one for changing the slides. Are you okay with this one? Yeah. Yes? So let's begin with the next conference that is called Can the ADP bioluminescence meter be used as an on-a-farm tool for assessing the microbiological quality of drinking water? And she's coming from Netherlands. Yes. Very welcome.
Well, thank you.
Thank you.
Wait a second for the slides, for the presentation to come up. Yes. Well, I'm going to talk to you today about a meter you can probably use on farm. So when you visit the farm, during a normal visit you do as a veterinarian on a farm, and you want to advise your farmer, how can you help him to get some more insights of what he's doing on the farm and how he's working with his animals, and how can you show him at the moment, at the time that you're there, that something is good or maybe can be improved. So what we did is we had this ATP meter, and we came across different water qualities in the field, especially for calves, and we wanted to see whether we could use this meter to say something about the contamination, microbiological contamination of the water, of the drinking water. Why is this so important? We all know that water is really important for cattle. Obviously to stay alive, because it's the main source of fluid they get, but also to optimize growth and production, and even maybe maximize growth and production, because they take more water. Sometimes it's proven that they take more dry matter, and as they take more dry matter, they can produce more. We also know that there's a lot of aspects to take in account if you look at quality of water. But something you can do on farm, and what the farmer has a lot of influence on, is the microbiological contamination. And up till now, it's still evaluated under laboratory procedures, but maybe you can do this on farm as well, so that the farmer can start immediately doing something about this. So we wanted to use this meter, this ATP meter, and we already know that we can use this meter for to evaluate the contamination of surfaces and also as liquids. So maybe we can use it in the water as well, and that's what we did. First, about the water, we had this study done in the US by Timothy Johnson, and it said these nice things. While milk is the primary source of water for the calf, consumption of additional free water is required to support optimal growth and health in the bovine. And further, that when supplemental water was not provided, this results in a 31% decrease in dry matter intake and a 38% reduction in weight gain. So for each extra liter of water consumed, there was a corresponding increase of 82 grams a day of dry feed intake and an increase in weight gain. And if you look at calves, and when they're in the first months of life, weaning is a really important step. They have to go from milk to intake of dry matter intake. And then the water comes in. So if they have good water at that moment, it's more probably that they drink more and that they also take more dry matter. And that the moment of weaning will not be as harsh for them because they already have more dry matter. So water quality during this period is really important. Well, we wanted to focus on the degree of microbiological contamination, and we used this study for it. It's a study from the Netherlands from 2013, so it's already a while ago. But we wanted to focus on this aspect you see on the top, the bacteria count, and as I said before, there are a lot of other things to take in account if you look at the water quality, but most of these things have to be done at the laboratory. But I want to focus now on the top one. And if you want to have suitable water for those calves, you have to have a degree of contamination that's below 100,000 cells. And if it's not suitable, it's above that threshold. Then the meter we used. As already said, it has been simple and very useful to evaluate the contamination of surfaces and liquids. But how does this meter actually work? So here you see how it works. What you have in the meter, you have this reaction between the luciferin and the ATP produced by bacteria. And what you see then is that the reaction with the luciferin, you get a production of light. So, as you see actually here in the photograph with fireflies, so the higher the contamination, the more light is produces. So, the more light you see. So, and that's what this meter is evaluating. Now, onto the study. Can we use this ATP meter as an on-farm tool to determine the bacterial load of drinking water? So what did we do? We had a total of 153 drinking water samples from different cattle farms in Belgium, and they were all submitted to the reference laboratory, the DGZ from Tornhout, Belgium, and these were all included in the study. And at the laboratory, all these drinking water samples were evaluated for the total bacterial cell count. And also, these were also swept with the aquasnap. You see it here on the background, on the right side. And we tried to see if there was a correlation between the total bacterial cell count and the ATP bioluminescence. And we wanted to try to find if we could see some optimal cutoff points for these ATP methods. So, again, what I already told you before, it's microbiologically suitable if the total bacterial cell count was below the 100,000 threshold, and it was unsuitable if this threshold is what we were measuring was above those 100,000. The count of 100,000. And then this is what we saw. On the left side you see the ATP value, and on the bottom side you see the total bacterial cell count. And you can see there's a correlation between those two. But that the correlation, there's still a lot of variation. It's not, you can see there are a lot of points around this line. So it's probably going to be really hard to get one specific threshold because there's a lot of variation. So we did some statistics on it, and we wanted to try to find what can we use in practice and what kind of cutoffs can we use out of these results. And this is what we found. So if you want to use 2 cutoff values, and again, as I told you before, the total bacterial cell count done in the laboratory was the golden standard here. So above the 100,000 was, below the 100,000 was suitable, and above the 100,000 was not suitable. If you then want to aim at the sensitivity for the not contaminated water, you should aim at an ATP value below 8. So that's really low. And if you want to have a specificity of almost 100, you should use the 650 ATP value. So that's if you use 2 cutoff values. Later on, I'm going to talk about what do you do if you are in between. But if you want to use only one single cutoff value and take some and take in account that the sensitivity and specificity is not 100%, but still pretty okay, then you can use the 15 value for the ATP meter. So this is again an overview of what I just said. So if you want a high sensitivity meter, you have to be really low, and the specificity is a little bit higher. And if you want a single cutoff, you can use the 15. So to take home, we can use this meter to evaluate the microbiological quality. And you can use that 2 value standard, so below 8 can be considered as suitable, and above the 650 you can consider as unsuitable. And maybe it's a good thing if you are in between that 8 and 650, you can send it to the laboratory to evaluate it once to do there another total bacterial cell count, or you can maybe repeat the ATP meter more often and see whether you still have the same result. I think it's nice that you can use this meter on farm and to give more advice and better advice to the farmers and help them to even improve on the spot. So that was my presentation, and I'm happy to answer questions if there are questions.
Thank you very much. We have enough time if you feel like for doing some questions. Nobody? Okay, thank you very much, please.
I wondered about, because the ATPase meter is not specific for bacteria, it's eukaryotic cells, right? So could you maybe measure things like crypto in the water and see how the ATPase machine is correlated to that measure, or am I barking up the wrong tree?
Well, I'm not sure.
Okay.
So I don't know.
Because it's right, isn't it? It's not specific for bacteria. It could
it's
be
the
any,
ETP that
any organic matter can make it fluoresce, right?
Mm-hmm.
So it could be parasites, maybe.
Yeah, maybe we can do
maybe
another evaluation and see
just
if we can find it.
Yeah, because the 0.5 is just moderate, right? The straight line that you put up. So maybe. Yeah. Just a thought.
Yeah. Okay, thank you.
Thank you very much. Any other question, please?
Hello. Was it a first or a second generation ATP meter that was used? I
am actually not sure.
All right. Not sorry.
I don't know. Yeah, no, I'm not sure. I
think the second generation ones are designed specifically for water use. Was it one of those?
I'm not sure. I don't think so. I'm not. Could be. Sorry about that.
Thank you very much. we do have some time if you feel like doing any other question, please. No? So I'm going to take advantage of this. Please don't go. Here in Mexico, we have some troubles for human consumption water. Okay. Is it expensive, this instrument for measuring, or it's not?
I think you have to invest one time in the meter itself. I think for us it was 1,200 or 1,300 euros. So, but then after that you only have to invest in the swabs. Okay. So then I think it's, it depends if you want to spread the investment cost of the ATP meter between the swabs, but I think the swabs are not that expensive. So then it could be an easy tool to evaluate the water quality.
Thank you very much. So this is all. We are going to give her an applause, please. Yep. And we have some free time. If you feel like staying here, we have 6 minutes until the next conference starts. And just for being sure about this, is Diana Patricia Martinez here? Bueno, pero nos esperamos un poquito a las 12:45. ¿Sí? Perfecto. Listísimo.
That's why whenever I come around, she's all over you. I know you want it. It's easy to say. And in the back of your mind, I know you should be on with me. Don't you wish your girlfriend was hard like me? Don't you wish your girlfriend was a freak like me? Oh, don't you? Don't you wish your girlfriend was raw like me? Don't you wish your girlfriend was fun like me? Oh, don't you? Fight the ceiling, leave it alone. 'Cause if it ain't love, it just ain't enough to leave a happy home. Let's keep it friendly. You have to play fair. See, I don't care, but I know she ain't gonna wanna share. Don't you wish your girlfriend was hard like me? Don't you wish your girlfriend was a freak like me? Oh, don't you? Don't you wish your girlfriend was raw like me? Don't you wish your girlfriend was fun like me? Oh, don't you? See, I know she loves you. I understand. I'd probably be just as crazy about you if you were my own man. So maybe next lifetime time I fall, I wanna hold you closer and look into your eyes. Remind me of the kiss we shared that summer night. Living in a movie, we can make a call. Oh, I can read your lips, and every time I fall, I wanna hold you closer and look into your eyes. Remind me of the kiss we shared that summer night. I wanna feel your heart, I wanna feel the beat. Anything for your love, I'd cross the 7 seas. When we're oceans apart, no, I will never leave. And even when it's dark, I'll give you all you need. Living in a movie, we can make a call.
So, let's start with the next conference, please. We are going to have, now from Mexico, it was time for us. She's Doctora Diana Patricia Martinez Albarrán, and she's going to talk about Do Goats Feel? An approach to sentience and emotions. So, we are ready.
Thank you for the introduction, Dr. Nuria. Good afternoon, everyone. So, first of all, I want to thank scientific committee to accept my paper. And this talk is very important for me because to arise the voice for all they cannot talk. So, nowadays, the issue of animal welfare and its impact on production and the final consumer is very present. Concern for animal welfare is growing and allows us to glimpse the feelings of non-human animals. So, to begin, it's necessary to define feel. This word refers to experience, to perceive something through the senses in a certain state or situation that can be external or internal. Sentience is not something new. Since time immemorial, and quoting a British biologist and naturalist, he says, "There is no fundamental difference between men and animals in their ability to feel pain, pleasure, happiness, and misery." Of course, we are talking about Charles Darwin. So, we talk about sentience now, and what a sentience is. De Gracia asks that there exists a stage in the development of humans and complex living beings where they become aware of themselves and their surroundings. So sentience is the capability to have positive or negative feelings. Sentience includes to have the cognitive ability to process and evaluate the actions of others in relation of themselves and the others. Remember action and consequences, generate and express emotions, evaluate risks, plan to achieve objectives or goals, recognize themselves and other individuals and relate, learning and choice, and for the last but not least, experience pain or pleasure. So, talking about pain, Snedden in 2009 defines the perception and aversive sensory experience of a nauseous stimulus associated with actual or potential tissue damage. Given that demonstrating painful sensitivity in non-human animals is not feasible, multiple lines of indirect evidence are required to imply pain. 70 specific criteria taken together suggest evidence of pain and suffering. These include protective functions, and for example, within the 70 criteria, goats are present as they are mammals. As an example, they have nociceptors, they have pathways to CNS, central processing in brain, They move away from the notions of stimulus. They have protective behavior, and etc. So, pain in goats. Well, goats have the ability to feel pain in a sensitive, like a physical or emotional way. And pain is an unpleasant experience to lead the individual to suffering. This response to pain is aversive, this being a way of alerting and distancing the body from the painful source. So the goat in this picture has a limb injury. She is in pain, and due to the position, it's evident that she's suffering, right? So not all occasions animals will display signs of pain, but we can recognize them through the change in behavior and posture, like vocalizations, moving away, turning towards to the source of pain, avoiding movement, and facial expressions. So we can see in the first image that the limbs are abducted, the neck is stretched, and the mouth is open. So there's so many changes or signs of changes in the position. We see in the next image, the last one is, as we know, as orthognathic position. So in the next image, the sheep, we can see that he raises his limb, right forelimb, and probably due to a lameness. So it's another sign of the animal is in pain. So for the first time, we can conclude that the goats are sentient beings with all the things that we already talked. But wait, we are not done yet. So we need to talk about emotions. So what is an emotion? It's an immediate and intense physiological reaction of a short duration to an event, a subjective reaction to the environment accompanied by organic change like physiological and endocrine change. Well, the emotions, they are multidimensional states, prepare the body for adaptive actions. They also have social and personal adjustment functions. They are ephemeral things that appear, disappear, and change. And there are classifications according to quality and intensity, or pleasant or unpleasant. In general, the emotions present the basic instruments for survival, learning, and adaptation. 6 basic emotions are described. And the first one, but not the most important, is sadness. It appears in situations of loss or separation, helplessness, isolation, and pain. It is a displacing sensation associated with negativity, but it promotes social approach and decreases the activity in case of they have injuries. Well, the next one, as we can see in the video, I will describe first. So it's achievement emotion, feeling of satisfaction. It promotes cognitive performance, problem solving, as well as learning, memory, and exploration. It generates positive attitudes, strengthens bonds between individuals, even interspecies. In this video, we can see kids are learning and socializing through play while having fun. Of course, this emotion is joy. Okay, and this, what emotion is this? It's fear, right? When danger is perceived, although sudden visual, olfactory, or auditory stimulus may provoke a startled response. Fear depends on a more complex analysis in which current sensory information is compared with memories of previously experienced events. Fear triggers an intense aversion response that causes goats to flee, fight, or freeze. Another one, this is a feeling of intense disgust towards situations in which the goat is unable to control events, either due to the actions of other individuals or change in the environment. It involves an aggressive and violent behavior and may be preceded by other feelings such as frustration, pain, fear, and anxiety. It prepares for self-defense and involves emotional memory. We are talking about anger, as you can see in this video. The goats, the hair is up, and they are trying to figure out space in this. Well, these goats were mixed, so they try to establish hierarchy. Well, the next one is, it occurs when faced with unpleasant stimuli, triggering, The next one, it occurs when faced with unpleasant stimuli, triggering rejection and escape behaviors. This is disgust or revulsion. And in this picture, we can see the taste of medicine is not exactly very pleasant for the goat. And the next one is neutral position, neutral emotional reaction. It is the fastest. It occurs in response to a novel, strange, and unexpected situation. It also fades quickly, clearing residual activity from the central nervous system to make way for emotions congruent with the stimulation or the situation. So we can see in the video, the goats are just rejection from the stimulus, and they then get back and see what is happening. Another video, it's just, at least goats are surprised. They don't run away, but they observe that caught their attention. So something is noisy in front, and these 2 or 3 goats, they just get a small surprise. It's not a fear because they don't run away. So we have talked about basic emotions. However, there are also another states called feelings or affective states, and which are another long and very complex topic that could lead to another conversation, and probably in the next Congress in Istanbul. But I want to talk about a little bit what is a feeling. So it's a brain construction. The feeling is a brain construction from the sum of emotions and preconceived concepts based on experience that, when repeated, cause animals to modify the environment, the behavior, and these are long-lasting. So the feelings, it's really a construction of all the emotions even. For now, I will just mention a few, such as suffering, empathy, hope, and frustration. And the goats are able to feel these feelings. So these terms maybe seem very philosophical, but science is working to understand the minds of animals and their interaction. with the environment that surrounds them. This talk is just an approach to a topic that, although linked to ethology, human psychology plays an important role in understanding it to determine animal well-being. So, and why is it important to know and recognize emotions and feelings in goats? Well, it's important by the animal welfare is nowadays very on the table, right? So it is important to know and recognize basic emotion as significant elements for the determination and evaluation, not only for the communication and interaction that exists between goats with their environment, their conspecifics, and other human and non-human animals, but also for addressing the problems of well-being and its relationship with environment. It has even been pointed out that well-being is not simply the absence of negative affects, but also the presence of positive affects. And considering them can change the way we communicate with goats. So this open to start many lines to research the emotions, the feelings, and the sensations in this gorgeous species. So thank you very much. If is there any question? Thank you.
Thank you. It was a very nice conference. So we have time for questions. Be my guests. Please. Thank you.
I have two comments. One is you mentioned empathy in the last slide, right? So I could see frustration part and others you can understand. How do you measure empathy in a goat?
Empathy, well, is hard to measure because there no exist researches that talk about the emotions and feelings in goats. But we can compare these species with the cow researches. And there's many researches about the facial expressions, mainly in pain signs. But yeah, it's a little bit hard because it's physiologically it's hard to demonstrate in humans. So in animals, it's more, you know. So, but they have a behavior that can, maybe we can humanize these actions or these signs, but it's probably behavior that we can to equal the behavior, probably with the other congeners. So, and it's very, ¿cómo se dice? Muy, very...
Difficile?
Difícil, yeah. Yeah, but this it's up to you. I mean, it's for each individual is very different how to see how to appreciate. I mean.
Thank you.
Welcome.
Thank you very much. Yes, please.
Hello.
Hello.
Thank you very much for this nice presentation. I personally don't know how to read all the feelings, you know, differentiate between. I can see when a kid is happy, it makes me happy, seeing them jumping around. But I know that in dogs and cats, you made studies of brain activity when they respond to different, and we interpret a dog,
Yes.
being happy. Then we can. Do you know of any studies in goats where we can actually see what is going on in their brain?
Yeah, well, they have different attitudes. One of them, if they have a connect with you, it's because you don't hit them, you don't attack them. So otherwise, they run away. But if they have a connect with you as a dog with you, it's easy to see when the dog is moving their tail. But in goats, they can approach to you. They have the capability to follow you even and make things that you can, like a training, but it's like with a gift, and they receive the response that it's positive. I mean, so it, as well, it's like different appreciated. It's hard to say that they have feelings, but they have a connecting with you in some moments.
Yeah, one more question was, is there any way we can measure if your interpretation of the goat is actually true? So can we measure dopamine, serotonin, or whatever?
Oh, yeah, of course.
And can we see the brain activity in the goats?
Yes,
But you
and
have not
even the
studied that
cortisol, and yes,
Yeah.
we can measure. We want, this is the approach, because we want to start lines for researchers to measure these hormones and chemics reactions. So.
Okay, thank you.
Yeah.
Thank you very much. I'm so sorry, we don't have more time, but you can talk to her. Okay? Yes. I'm so sorry. We are going to the next one, that is Antini Mersha. Yes. Are you here? From Ethiopia? No? At one, no, at two. Well, if you feel like we can wait for a minute, otherwise we are going to play a video. It's a very nice video, I recommend it, so if you're feeling like staying, it will be a pleasure. So let's start with a video. It's a very nice video. If you feel like listening any other conference, we have another rooms. So if you feel like changing, it could be a good time just for getting the starting of the next one. Or if you feel like watching the video, but I'm so sorry, it doesn't have any audio. So whatever you want. or if you want to get your place for launching.